All right. Well, we're back. So thank you for bearing with us, but we got everything rebooted and we should be good for the rest of the dive. I think we've still got about 6 hours left. So enjoy. I'm going to hand it over to Jimbo for a little bit here, and I'll be back later.
That's the way to do it. If I leave periodically,
Sí. Claro. Sí.
Is there a background current on top of all this too?
Sí.
Or is it? No.
Sí.
Yeah.
Ah. Sí. Ah, qué interesante.
So I don't put them together as just 3 separate photos, but the mirrors get used to talk.
Is it laser? Him?
But the middle lens, I have a wide.
Ahora vimos que volvió. No sabíamos, había caído la internet, se había caído el streaming. Pasa en todos lados, pasa también en el Falkor 2.
All right. We're refocusing. Back in search mode.
Estábamos atrás de, bueno, ese puntito rojo que estábamos viendo antes en pantalla es un anfípodo. Pero creo que bueno, lo dejaron pasar y ahora vamos a seguir hasta los 1200 metros de profundidad. Todavía tenemos varias horas por delante para seguir encontrando más cosas. No, un quitognato.
Oh, something just swam by to the right.
So the more tether we have, the better, you know.
Oh,
You got another.
You already see
in
that?
the middle.
Here, I thought I was the discoverer.
Keep going, keep going.
Keep.
Russ is gone.
Rest is gone, don't worry.
Nothing to see.
Another fish situation. And maybe a self case.
Just the perception of being in this room.
Is this Doliolus? No, it's praying. It's a prayer tail with special swimming bells. They're the ones that you were liking, right?
Wow.
Are they tethered?
It's a praying.
Oh, lo que estamos viendo
Probably
ahora
not rosacea.
es un
Too much
sifonóforo
red.
que eran coloniales y se quebró, entonces
Yep.
estamos viendo todos los individuos por
Moving
separado.
on, moving on.
What kind of iphonophore do we think this was?
It's a calicophoran, a prayed.
Yeah. Cormedial bells.
Yeah.
How's that for an engineer?
Yeah. Not
I have a
bad.
lot to learn, it seems. No, you're in it eventually. It's taken me a while.
Your tall poppies like
Dougal, where are the promised rhizarian soup?
What are we now? About another 50 meters deeper.
Remember?
Yeah.
Yeah, they're always deeper.
They should be kicking in soon.
Now it's just when you see it.
So Mandela by
Oh.
tentaculata.
Oh,
Moving on, moving on.
Moving on, moving on.
what I meant.
Agora uma medusa do grupo de narcomedusa,
Oh,
animal que tem esses tentáculos acima da
It's a
umbrella, né?
narcomedusa.
O
So
tentáculo ele fica na
Mandela
frente,
by
na
tentaculata.
direção da natação, né? Então, a primeira parte do corpo do animal que chega quando ele está nadando é o tentáculo. Isso ajuda ele a se aproximar de presas sem ser percebido. Uma adaptação importante para um tipo de predador de copépodes, que são animais muito sensíveis. Então ele vai avançando e com o tentáculo à frente se aproxima primeiro da presa sem causar vibração.
Yeah, is that yeah, what, how did that get set?
Ah, that's not that hard. I mean, 1200 was just like the dives are said to be explained.
I would.
Just so we can say we looked at the entire water column, I'd say.
Set that as a,
Yeah.
we could
I
take
don't
that.
know if we wanted a CTD reading all the way down.
Well, at 1200 we're supposed to shut a bottle,
On a flat coast
Yeah.
that.
a Niskin bottle.
Yeah.
And collectivize area in it.
In it, right?
Yeah.
So we don't use one
So
of our
that's gonna be a
canisters.
tricky,
I
that's gonna be a tricky
Or not.
maneuver.
I searched
there's a little
and they're
filter
not
pad.
there.
Is it a house?
Like a
Up
blue
above.
day.
It's super small.
Yeah.
Oh,
Oh,
another
blue.
one.
Room.
Another one on the left.
Houses, like a neighborhood.
Is there a preference?
The left.
Left? Yeah.
Like a bird house?
Neither.
Oh, that's a hot place.
What's the
Distinguishing
distinguishing feature of an Florida? Is it the double lobe,
This is a filtration
Yeah.
structure it's secreting.
like the lung shape?
It's a very, like the lung shape, and it looks really smooth compared to a lot of the other lavations.
Animal
Now
there?
this
No.
one's smooth, there's no clubs on it.
Oh, what?
It's got
Can
some
we
pleating,
zoom in
huh?
a little bit more?
It does have some
It does have a
pleating.
lot of pleating, which reminds me of a different one.
Look at their own glasses on it.
Uma coisa, o link pro YouTube cada hora é
He's good in the shadow cam.
cada
That's more like
This one's completely smooth. So this is like a pleura. It's a
What's
genus.
pleura?
That's a larvation or appendicularian. I don't know what you have in there.
tunicata, I think, lavation. Oh, it's a good shot from the side.
Yeah.
It's got that L-shaped thing
Yeah.
happening there.
So you're saying like
Now
a
that should
pleura.
be the animal right there, is that?
That's there. That's
Yeah,
the trunk
that's the
there.
head.
Oh yeah.
You can see the
there's a
tail
tail.
is like
Yeah, but it's not beating.
This
No.
looks really batho-like to me.
Well, we just scared the heck out of it.
It's still in there.
Yep.
That looks a lot like.
it's too small.
That'd be really mean.
Nice shot.
That's
Really
great.
beautiful.
What's that blue taste at the back?
That's the tail.
Iridescent.
Tail,
Iridescence
it's
of the tail.
refractile of
No,
the tail.
the tail. It's the very further back. Like 3 days.
That's great.
You see the 3 dots
there's
beyond the head?
the money shot.
Those are the pre-filters on each side. The tail is silent.
This
It's
is the
not
tail.
I see the tail, but
That's
okay,
a that's a nice looking house.
Yeah.
It's interesting, it's fully inflated, but he's not pumping.
Boa noite, pessoal. Então, estamos vendo aqui um larváceo, né, que é um animal
Siesta time.
do grupo dos cordados, ou seja, ele é um pouco mais próximo na árvore evolutiva dos vertebrados, ou seja, da gente. Então é um animal que produz uma casa de muco, né, ele secreta muco por uma estrutura que se chama endósteo, que tem a mesma origem evolutiva da nossa tireoide, né? Então ele produz um muco
I'm taking photos of the
e
photos.
essa casa
It's pretty meta.
serve para filtrar partículas, né? Então essas partículas são absorvidas e o animal ingere essas partículas, né? Então, através dessas estruturas que parece um arco, as partículas pequenas ficam retidas e às vezes quando esse filtro fica entupido, o animal deixa a casa e produz uma outra casa dessa, né? E assim essas casas que vão sendo produzidas sucessivamente acabam afundando e são um dos grandes contribuintes do que a gente chamado de neve marinha, né? Que são as partículas que vão descendo e abastecendo os animais que vivem no fundo dos oceanos.
So what black was presumably. Yeah, I think there's less carbon at least relative to the animal.
No, the beer has no bubbles, only the animals.
La profesora Cecilia Morgan está ahí nos indicando que no es una medusa, que es un larváceo.
What happens is that would also be okay.
Can we push
Hola
forward a
Cecilia,
little
acá
while
Silvina.
going down, please? Still got that blaze like wonderful tentacle hanging off the top of the camera.
And you can't quote the animal here and say that
That's
it's
like
good.
monophores kept up in its swimming speed with its whole dive.
I don't think it'll have any effect to the animal. I could just soak the animals in BSA.
Como está mencionando Cecilia ahí en el chat y nos contaba acá en portugués el profesor Renato, el larváceo es pariente lejano pero pariente nuestro y produce esa casa de muco que estamos viendo en la pantalla, esa casa transparente.
The slip boundary is still, it's not as
Prolegomna is a cytophonophore.
Yeah. Similar to the one we saw earlier.
Continue.
Yep, keep diving.
I think we can hit
This
1,000
room
meters.
is really tough.
It's like meh.
If you
Meh.
expose your interest,
Oh,
then
what
it's
kind
a
of-
tough room
Oh,
to
never mind, ignore it.
Yeah.
Careful.
Oh, yeah.
Medusa?
Oh, la medusinha.
Oh, watch your Nemo. Is there a Nemo? Yeah, I reckon.
La medusinha.
Opa, agora encontramos um dos animais que é de interesse aqui da expedição.
Well, one of the clades.
Um animal que pode ser uma espécie desconhecida da ciência, um animal que pode ser uma espécie nova,
It's a Trachymedusa halopeatidae.
da família Alocreatidae, né? Uma espécie bem bonita que tem um ápice ali pontiagudo, como vocês podem ver,
It's to break out of
e que parece ser, Alicreatidae,
of the
a gente vai chegar mais perto, parece bem interessante, vamos ver se realmente é o que pensamos ser para fazer a captura do animal.
There you go.
También les recordamos que, bueno, a bordo hay unas investigadoras que están secuenciando en tiempo real en cuanto estamos en el crucero y varias de las especies que no se sabe bien, o varios de los organismos que no se conoce bien las especies, se está secuenciando el genoma completo.
Just get one nice close zoom so you get the morphology. Don't have any morphological data for South Atlantic.
Não sei que você queira nadar a bem médio.
Esa es peligrosa.
Wow.
It's got the same
Todos os
slight
medusas têm
shoulder
veneno,
as the Pacific one does.
mas nem todas têm venenos fortes.
Pointed one like the Arctic variant. Oh.
Uma coisa que é bem interessante, assim, é que na maioria das vezes que a gente faz esse tipo de estudos, né, a gente obtém os exemplares através de coletas, né, seja por rede específica.
Because they have a policy
E aí,
to
quando eles chegam na superfície, ou eles já estão muito detonados, né, estão rasgados e tudo mais, né, ou eles são fixados em produtos especiais como álcool, formol, e vão para coleções, né, e perdem a cor. E então, com esse tipo de equipamento, a gente consegue ver um pouco do comportamento deles e também consegue ver o padrão de colorido natural deles. Às vezes eles produzem bioluminescência, às vezes eles produzem fluorescência, como a gente vê no caso dos quitenóforos. E essa cor também, quando vai para os museus, para as coleções científicas, acaba perdendo. Então é uma oportunidade também da gente conseguir observar o padrão de colorido deles. E, obviamente, um pouco do comportamento também, né?
Hola Sole, gracias por acompañarnos acá en el streaming.
Was that on the lower right?
Complementando lo que Marcelo decía, de que gracias al ROV podemos entender mejor los movimientos y estudiar bien el formato y los colores de estos organismos que cuando llegan a la superficie ya están bastante destruidos. El ROV de esta vez fue equipado con una serie de cámaras iris que lo que hacen es sacar imágenes continuas a lo largo del animal como si fuese una especie de máquina de cortar fiambre, y todas esas imágenes se van acoplando para formar toda una estructura tridimensional y poder tener bien la estructura del bicho. Y además con estas cámaras también podemos saber el volumen, y al estudiar o medir las corrientes que pasan entre los tentáculos o entre la superficie del animal, se pueden estudiar y mejorar los conocimientos sobre los movimientos de estos organismos, que muchas veces, como dijo Marcelo, cuando llegan a superficie es más difícil de estudiar, porque se tienen que aclimatar, se tienen que poner en diferentes acuarios y obviamente no es el hábitat natural. Ahora lo que tenemos en pantalla es un ctenóforo.
And what would the tax be for that?
Ctenophore, or oh,
Lampeteis. Oh, we collected one in the deep sample, that's right. There we go, that's a good shot. Okay.
Would this be a lobata?
It is, yes.
Oh, qué lindo.
Agora estamos vendo esse quitenóforo lobado, né? Ele tem essas duas estruturas arredondadas, que é por onde ele cerca o seu alimento e ingere o alimento, né? Mas uma coisa que se destaca nesses animais é isso que vocês estão brilhando ali, né? Parece uma luz, parece uma coisa que o animal está emitindo, mas não é uma luz que vem dele, né? Na verdade é a luz do ROV que está iluminando ele, e ele está refletindo essa luz para a gente de uma forma um pouco diferente, né? É o mesmo processo que ocorre quando você vê um CD ou uma gota de óleo no chão, né? Que em português pelo menos se chama furta-cor, né? Então isso é um furta-cor, que acontece uma refração seletiva das ondas que incidem sobre um objeto, e é o que a gente está enxergando ali agora.
No. Four.
Uma coisa muito interessante desses cruzeiros é que é uma oportunidade para a gente também interagir com diferentes áreas do conhecimento. Então aqui a bordo tem pessoas, né, assim como a gente está na universidade, né, professores, mas também, então entre a gente tem oceanógrafos, biólogos, de diferentes áreas, pessoal mais ligado à taxonomia, tem outras pessoas mais ligadas à fisiologia, à neurologia também. E tem uma equipe grande de engenheiros que desenvolvem esses equipamentos. Então, muitos desses equipamentos foram desenvolvidos especificamente para trabalho de pesquisa biológica em águas profundas. São equipamentos que são...
To make the composite, you just find a common orientation and superimpose all the different scans on it, or one scan. You're always looking for just perfect scan.
Esses equipamentos eles precisam ter uma resistência muito grande à alta pressão. Então, esses scanners, essas câmeras, todas tem essas caixas estanques, né, que conseguem resistir. Agora o ROV está a 1.040 metros, mas esses equipamentos têm capacidade até o fundo dos oceanos, e em alguns casos mais específicos até as grandes fossas, que tem a pressão hidrostática, ela aumenta bastante com a profundidade, aumenta 1 atm a cada, e consiga desenvolver o trabalho lá embaixo. Uma outra coisa também muito legal é que aqui a bordo tem uma artista, ela está até aqui na cabine com a gente, é a Young, ela é coreana, e ela veio atrás de inspiração, para se inspirar nesses organismos para o trabalho dela. Então essa interface ciência, divulgação e arte é super importante também. E agora a gente está vendo o ctenóforo cruzar o laser, né? E parece que ele está pegando fogo. É uma coisa muito legal de se ver, assim. E realmente é uma coisa que, mesmo pra gente na academia, que trabalha com ciência, assim, eu nunca tinha visto isso antes, assim. Então é uma coisa muito legal de se ver.
Like a low rebate? Yeah. Like I thought like baked fish.
Right.
Okay.
We do now. When I looked at it, I probably got close.
Lobular.
Yeah.
Okay.
Mira, have you played Dragonverse yet? No. Why no?
Next on my docket.
Yeah, that's a good crash course.
Do that tonight and then come in tomorrow and just be gone.
And you'll have all that.
Na cabine com a gente agora tem 17 pessoas, entre o piloto do ROV, os operadores, né, que controlam os braços do ROV, o pessoal da pesquisa, tem a Yang, que é essa artista que está com a gente aqui, e tem também o pessoal de divulgação, que cuida da página do Instagram, né, e tem também os 2 engenheiros que desenvolveram esse equipamento. É uma equipe multidisciplinar, né, e altamente especializada nesse tipo de ambiente, assim. Porque ninguém faz nada sozinho, a gente precisa dessa ajuda para desenvolver tudo isso. Então as pessoas dão as ideias e eles desenvolvem de acordo com as necessidades do tipo de ambiente, do tipo de projeto. E particularmente esse cruzeiro tem uma dificuldade a mais, que é justamente explorar esse ambiente pelágico. Quando nos outros cruzeiros que foram feitos na Argentina, no Uruguai, o ROV explorou um ambiente demersal, né, um ambiente que é próximo do fundo. E isso faz com que o ROV os organismos fiquem presos em um lugar, fiquem parados. A gente pode ver aqui que os organismos estão se mexendo o tempo todo, assim. Então tem que ter um controle muito fino do ROV, né? Esses pilotos de ROV, como a gente conversou com o Léo, né? Eles são super treinados para isso. É o mesmo tipo de treinamento que se dá para a área da indústria, né? E eles têm um controle extremamente fino do ROV, assim, a ponto de né, conseguir parar o ROV no meio do oceano, se mover de acordo com as correntes, para que, né, esses bichos super delicados possam passar pelo peixe de laser ou possam entrar numa armadilha dessas sem ser danificado. Então isso é legal também porque é uma área assim, para quem pensa em seguir carreira, né, na oceanografia, na biologia, na engenharia, isso é uma área que está em uma expansão muito grande, principalmente com a expansão agora, né, da exploração de petróleo em mar profundo, da mineração de mar profundo. Tem um campo de mercado que é bastante interessante. Além dessa parte da biologia, que é o que move a nossa paixão, né, tem também essa parte comercial de conservação das espécies, que é um campo super importante também.
And for low baits, are they usually low
Okay, Killy line?
Eu estou vendo algumas pessoas agradecendo aqui o Schmidt, né? Esse instituto é muito legal, porque é um instituto com essa devoção assim, para dar oportunidades para as pessoas fazerem esse tipo de pesquisa, porque é uma pesquisa muito cara, né? Os equipamentos são extremamente caros, um navio desse é extremamente caro. Para vocês terem ideia, a nossa equipe aqui tem 70 pessoas a bordo. São 23 pesquisadores, né, e os demais são tripulantes, são pilotos de ROV, tem o pessoal que trabalha no comando do navio, tem o pessoal da cozinha, o pessoal da faxina, tem o pessoal os engenheiros que trabalham na casa de máquinas, né? Então não é uma atividade muito barata de se fazer. E no nosso caso aqui, a gente tem o custo dos equipamentos, dos pescadores que vieram, mas eles não cobram nada pelo uso do navio, nem pelo uso do óleo, que são coisas que às vezes são fatores limitantes para esse tipo de pesquisa. Também esse ROV ultramoderno, é um equipamento caríssimo, que é difícil ter acesso a isso. Às vezes as grandes empresas petrolíferas têm acesso a esse tipo de equipamento, mas isso não fica disponível para as universidades, para os pesquisadores. Então essa é realmente uma oportunidade ímpar de fazer esse tipo de exploração, né, científica, sem ter esse viés comercial puramente pela paixão de descobrir coisas, de explorar organismos, de explorar áreas novas. Isso realmente é uma oportunidade muito bacana para quem está aqui a bordo.
The scan doesn't care which direction you go, right?
Does care if it's like
Okay. There's my phone.
Oh, I see.
so if it goes
If there's any along plane.
yeah.
Turn that one.
Now, if this was a sphere and they'd be using the correct. There's not a lot of
Yeah.
spherical only in physics groups.
We got one in the middle.
Right, the spherical cow.
Exactly. It's a big one. So ours can stand with me. Last one.
Okay, Killy line. Okay, turn it up. Far to the right.
Bless you. Again, yeah.
There's somebody on the phone. Put it out to 7.
Estamos demorados tanto en este lugar porque estamos intentando poder escanear este tenóforo y para eso necesitamos que esté lo más quietito posible, que haya menos corrientes para poder tomar imágenes desde diferentes ángulos y poder reconstruir la estructura.
I'd give you an A.
Or I'd take it.
We'll take it. We'll take it.
But I've always been an easy grader. There are others here.
Recordando que ahora a bordo del Falkor 2, durante esta expedición tenemos por objetivo estudiar toda la biodiversidad zooplanctónica de media agua entre los 200 y los 1200 metros de profundidad. Esta región tiene muchas especies que nunca antes habían sido estudiadas y esta región en sí no es muy conocida. Entonces todo lo que salga de estos resultados obviamente van a incrementar todo lo que se conoce de la región del Atlántico Sur.
Doing great. All right. Keep diving. 20 meters from being a kilometer deep.
Pessoal, eu estava conversando com a professora Kakani Katija, que é do Monterey Bay Aquarium, né? Eles desenvolveram esse equipamento de escaneamento que vocês estão vendo, né? Que se chama DPIV, né? Então é um tipo de câmera que tem uma iluminação a laser, né? Então é um laser vermelho que gera um plano onde que ilumina o animal, né? Então essa câmera faz 60 fotos por segundo E à medida que o animal passa pelo laser, ele vai fazendo várias imagens ao longo do comprimento do corpo do animal. E essas imagens são depois reconstruídas por um programa que faz um modelo tridimensional do corpo do animal. Então é um método muito sofisticado que faz um escaneamento, né, tanto da superfície externa como interna dos animais, e precisa ter uma passagem do animal perfeita pelo plano do laser, né? O animal não pode rodar, tem que ficar o maior tempo possível dentro do plano para fazer um bom escaneamento. E além da morfologia do corpo dos animais, é possível capturar o movimento dos fluidos, né? O movimento da água que o animal faz ao nadar, né? Então é possível também entender como que funciona, né, o corpo desses animais para natação, né? E essa informação sobre como funciona o corpo dos animais é algo que é de particular interesse, porque o pessoal dessa instituição, Monterey Bay Aquarium, eles têm um laboratório de bioinspiração, né? Então eles usam modelos animais para gerar inspiração para robôs, por exemplo, para propulsores que são colocados em veículos aquáticos, né, que usam animais como inspiração, né, para se desenvolver essas mecânicas.
It's Claustrophes, the genus is Claustrophes. There's several species, and we're getting close to seeing its analysis there.
Respondiendo a la pregunta de Julián. Hay varios investigadores a bordo del barco, cada grupo con diferentes objetivos, pero con un objetivo común, que es estudiar la biodiversidad mesoplanctónica. El grupo que estaba contando Renato hace un ratito, de la profesora Kakani, ellos están estudiando esto justo con el ROV, que pusieron este sistema de cámaras para escanear los organismos y poder también estudiar los movimientos. Con estas imágenes y con estos estudios, el laboratorio de ellos se inspira para estudiar y crear nuevos aparatos para estudios de robótica que pueden adaptarse. Después tenemos varios investigadores especialistas en crustáceos como krill, copépodos, anfípodos. El profesor Renato Nagata es especialista en nidarios. El profesor Marcelo, de la Universidad de San Pablo, trabaja con peces de mar profundo. Entonces todos acá estamos con así diferentes objetivos, tanto Silvina como yo, si bien nuestro background es en mamíferos marinos, ambas trabajamos con isótopos estables, entonces todas las muestras que vienen en las redes o en el ROV, juntamos algunos especímenes y los guardamos para posteriores análisis de isótopos estables, lo que nos permitiría reconstruir cómo es la red trófica de esta región.
O pessoal que está online, se vocês tiverem alguma pergunta para saber da gente, assim, da carreira da gente, como que a gente fez para estudar esses bichos, o que é nosso interesse, fiquem à vontade também, tá? Podem aproveitar a oportunidade, que eu sei que deve ter bastante gente que tem interesse em seguir carreira, ou tem alguma curiosidade, então fiquem à vontade para perguntar, né, sobre as nossas carreiras, como a gente o que a gente faz, onde a gente ensina, que tipo de pesquisa a gente faz também.
They can have any calycophorans can have anywhere between 1 and about 6 more bells. So just Voltaire and Hippopodius, those like pine cone pineapple things are calycophorans as well. They don't have a float though.
Eu falo isso também porque a gente normalmente quando vê esses vídeos, né, os livros, né, falando sobre esse tipo de fauna de mar profundo, a gente não consegue perceber que isso faz parte da nossa realidade também, né, que países como Brasil, Argentina, Uruguai, Colômbia, Chile, né, o mar profundo é muito importante, né, faz parte da zona econômica exclusiva dos países e é um campo muito importante também.
Yep, Halascaroconica. That's nice shot of it. Its feeding posture.
Get your lights.
So another
Below
producer,
on.
right? You know,
There we
if
go.
you stay off that, and then we just did one like a single shot scan with all of the tentacles around it, that would be
cool.
Really cool visualization.
Halascaroconica, it's a Trachymedusa. We're good now, I think. We got the fishing posture well, and I can ID it, so we can keep going.
1,000 meters!
100 deep.
Estamos celebrando que llegamos finalmente a los 1200 metros de profundidad.
1000 metros.
Ah, 1000. Todavía estoy dormida. Llegamos a los 1000 metros de profundidad y nos faltan 200 más para después empezar a subir.
So I reckon from that fishing posture that the definition that says they're in a single row has got to be wrong, because they wouldn't be doing that if they were all in a single row.
You mean on the margin?
Yeah, on the margin.
No oceano, 1000 metros é uma profundidade importante, que é mais ou menos quando a luz solar desaparece completamente. A gente divide as zonas oceânicas na zona epipelágica, nessa zona ali da superfície. Apareceu um peixinho ali agora.
Might be a fish. Let's go down and have a look. Nemodian.
Vamos ver o que é isso.
Our first Nemodian of the cruise.
I thought it was a nimmer.
A flying
Ah, não é um peixe, é um
Flying
nemertino.
cow's tongue. There we go.
Ooh, this would be a really good iris.
Wow.
They're actually called
É um invertebrado.
Cartilages.
Quer falar mais sobre Renato?
It looks like a fish, right? No, it's a Nemertian one.
Renato.
I mean, everything's a worm, but
And when we say worms, we mean
Nemertíneo
This
é
is
um
not an
animal
annelid.
bem interessante que na árvore filogenética dos animais está bem relacionado com moluscos, né? Então é como se fosse uma lesma que
no,
flutua,
there's
que é
white-
plantônica,
there's
né?
white
Que tá
ones, there's
nadando,
black ones,
desfilando aí na
there's
nossa
big
frente.
ones. This doesn't look orange enough to be.
Hermetíneo pelágico.
I just wanted to see it where the taxonomy.
Bem interessante porque não é algo que é muito comum, né? Que os nemertíneos sejam pelágicos, são animais mais bentônicos, mais associados ao fundo.
Yeah, I'll open them up after.
Então, como eu ia dizendo, né, a luz solar ela penetra até nas regiões oceânicas, onde a água é muito clara, até mais ou menos 200 metros de profundidade, com uma intensidade suficiente para fotossíntese. A partir de 200 metros ela já é uma luz muito tênue e a fotossíntese desaparece completamente, mas ainda há penetração de luz solar. E depende da região dos oceanos, mas normalmente, né, essa luz consegue penetrar ainda até 1000 metros de profundidade. A partir de 1000 metros é uma zona onde a luz solar não penetra nunca. É o que a gente chama de zona de escuridão total, né? E a gente percebe que começa a mudar a fauna também, né? Tá aparecendo muitos organismos que não apareciam no meio do caminho ali, quando a gente estava descendo.
Is that autofocus now? Yeah.
See how close that's at? It's like you to expand my light array.
Temperature, light cycle.
É um modo.
The light array will be expanded. Exit. Relax.
Oh, is that a still misses?
It moved. Scared me.
Outra coisa que a gente consegue perceber também é que nessa região a água é muito fria. A temperatura da água agora está em torno de 4 graus Celsius e a gente está muito próximo do Equador, onde as águas superficiais são mais quentes, mas a essa profundidade os oceanos são mais ou menos homogêneos. Então, independente de onde se esteja, se é mais próximo do Equador ou em altitudes mais altas, a temperatura da água vai ser a mesma em praticamente todos os lugares do mundo, com exceção, obviamente, das regiões polares, né? Que aí realmente a água é muito mais fria. Ela pode chegar próximo de zero e, às vezes, em alguns lugares, ela pode ficar até levemente abaixo de zero, sem congelar por causa da pressão. Mas é um ambiente, então, sem luz solar, com altíssima pressão hidrostática e com a temperatura muito gelada.
Did it record?
Back in 5 minutes.
That was Halifax minimum, probably.
Esse é o tipo de ambiente que a gente chama de ambiente extremo para a vida. Então, esses organismos que estão nessas regiões, eles estão perfeitamente adaptados para essas condições. E muitas vezes, quando eles sobem no ROV dentro dessas garrafas, as garrafas são pressurizadas, mas à medida que o ROV vai subindo, a temperatura vai aumentando. E essa temperatura mais alta mata os organismos. Então, isso... Eles têm essa capacidade de ver águas muito frias e eles têm uma baixa tolerância a temperaturas mais quentes.
Because it's got 3D structure, it's got those knobs on it, don't it? I guess you probably already scanned it.
Uma outra coisa que é curiosa também é que alguns desses organismos que vivem em águas profundas, aqui na região dos trópicos, né, em altas latitudes eles conseguem viver águas um pouco mais rasas, que é onde tem águas mais frias, eles acompanham as massas d'água. Então tem alguns grupos de peixes, por exemplo as quimeras, que no Brasil são bastante raras, né, e na Argentina, por exemplo, elas são pescadas em águas bem mais rasas, né, e viram até recursos pesqueiros. Isso no Brasil não acontece, assim, aparecer uma quimera é muito raro e quando aparece é no sul do Brasil, assim, mais para que nessa região elas estão em águas muito profundas onde normalmente os pescadores não têm acesso.
Well, I saw you had lots of particles. Were you just doing heavy gradation line?
There you go.
There you go. Yeah.
It's not in the original technical department anymore.
So why didn't that get more?
You can't get it from the side.
You see that this is a hotkey too for record, but it's like Command Shift plus R or something like that. Six bucks up.
There he goes.
Say again, yes.
Oh, there's a nice magenta syndicate there.
But it's like shift command R or like it's never something you're
Focus.
gonna do quickly.
Where's the gradation line here?
What they mean to say is helpless.
Are we okay with that now? Or?
The language that's used in
It's German engineering.
Heating nav.
Figure this advance, huh?
No, I think I denied.
Or oh wait, no, we restarted. Can you look at what the threshold is for the event? Or did you set it? Because I've been setting it to 0.5. Oh no, there's no depth threshold set at all. So there's no event trigger.
We move on to something, or you still?
So it swam kind of through it? I don't think
That's
so.
earlier, right? Oh, I don't know that I did set up the event.
Well, I need to redo all the settings.
It's back to normal shape
I've also
again
done
now.
that already at least once. But like the event settings.
That's it. Come on, come on, come on. Is that a good scan? Is it good?
I feel like it should be. I gotta figure that out.
Okay. You find something that's more new?
There's another heating nav.
Dougal's not impressed with this animal. With this animal.
See it right in front.
I think at the top is.
Yeah,
Add rule.
that's been kind of hanging out there for a while.
Depth map coverage. Right ratio to 0.5. That's all.
Would it work?
Yeah.
Let's see.
There you go.
Hmm.
Was that 0.5? Was that 50%? The view needs to be there.
Yeah, we got it
That's
already.
good. Nice.
But how do we turn that into something that looks like what was just the background over there? On the Sierra Connect.
So moving around up there.
An octopedema, it looks like.
Give me something hard. That's why it's I really lost that flying tongue.
It's not for no reason.
Never
Right.
seen it.
That was just lighting up. That was lost.
All right.
Did you call the flying biltong or something? Never heard that before.
It's basically pseudo
Are we okay?
So
We're good?
what-
All right.
All right.
Let's
Maybe.
move the arm away so it doesn't hit the other one, I guess.
The photos I was taking of that one this morning, it's like an eyeball.
Yep. That was Helicreus minimum. Yep.
with spines.
Yes, yeah,
Like
it was
little
fine.
light switch
Yeah.
chain.
Pretty crazy.
If it works.
Bulbs, bulbs.
Mm.
Like warts.
Thick rods
So
on
rotate
yeah,
clockwise.
ends of the tentacles, yeah.
Rotate clockwise over there. Off. And okay, shoulder tail up over there. elbow in all of that. Elbow in, down. Rotate. Clockwise.
Red light district here.
A little more. Right, and then elbow down a little bit.
Yeah, that's good.
And freeze it.
Like it?
Nah.
Locked.
Too early.
Cool.
Fear of just.
That's pretty amazing. Just
Makes me feel like I'm in Amsterdam.
Multiple eyes.
here we go. We went back up 10 meters. We got some space to catch up.
We're no longer at 1,000.
I know. It's embarrassing.
So you live below sea level, so.
I actually, would, I would have lived on the beach.
Oh, yeah.
I was a foot above or something like that. Like right about 2/3 into the country where it starts to slope just above sea level. So if all the dikes broke, we were
Another keto?
our property
Oh, sorry. I was looking at the speed.
value would have gone way up.
Don't catch us beach
Yeah.
because you had that 1 meter of
Oh
elevation.
no, I was good with that.
Exactly. High tide run dicey.
You weren't perpetually like digging and then like.
Yep. I'm feeling good about this.
Slow down.
But so far, the only people found that I found is
Open the tin. Can we open
So
the
I
tin?
gotta find it.
Disable.
I did review.
So when you're going ahead and diving slowly, is the boat following?
Yeah.
Could we drive forward a little bit more as we're descending, please?
Okay.
And maybe pan the camera a little bit more right so we don't have so many machines in it. Thanks. No, I don't have anything against the machines. They're just taking up my visible volume.
I'm just scared of that. Like, just think of how many pixels we've recorded and how many pixels have been the same thing. Turn this.
And the same thing is like the pirate zone.
Coming up.
There's
Salt.
looks like a salt. Yeah.
Solitary salt?
what it is. Don't need to stop.
Yep.
What's it doing down here but eating marine snow?
Oh no, it's already, yes, sir.
Okay, moving on. Thank you.
Move on. Yep.
Yep. Nothing to see here.
Just another creature living at 1,000 meters beneath the surface.
Straight without seeing your red light.
Nothing we haven't seen before.
Right,
is what the scientists
2017, nothing to
say.
see.
Regular people, I have never seen one of them before. Like, hey, it's just a fish without a backbone. Oh, something in front of us. Cresota milzi, maybe?
That's something new.
It looks to be Cresota milzi. Get a zoom in before it realizes we're here and pulls in those tentacles.
So everybody hold the breath.
Hold your breath, hold your breath, hold your breath.
I mean, what if we did a scan, had one go at scanning this stuff?
So what exactly is this? Is it Cosildum?
Cresota milzi.
Put your laser on.
Turn the laser on. We got one shot.
Okay.
And you're recording this?
I am recording.
It's a Trachymedusa.
Oh, right.
It's still in its posture.
Can we dim the ROV lights when we do it?
Right.
Just want to dim the lights when we do it. Just get the red light on.
Put your light back on.
Turn up the music.
No, the
Yes, too.
outturn.
So,
Something with a little beat.
Selfie coat.
Okay, well. What's that little guy?
Okay.
Yeah, I don't know.
See what
So,
we can
yeah.
find. Punching slowly forward and down. Eyes peeled. Only stopping for copepods, right?
We're stopping all the way down. Every meter.
Oh, what's that in the top corner? Some kind of
Peep.
protist? Or a thing?
Rosarian, but not the one we're after.
Was it a protist though? Or is
It's a
that not?
protist, yeah.
We're
Rosarian.
accused of deceiving people about finding the.
Yeah, where are they all?
All these areas are protists.
Rosarian or protists, yes.
Oh,
There it was again.
do we need to stop and hang around it?
Yep. Let's go.
Yep.
We're looking for the guy with the forearms.
No, pues me encontré uno con cable.
Otherwise, Manu will have his forearms around my neck. Focus a little bit further out, please. Okay, a little bit further going forward. That's it. Past this left on the four. Past the cook pod, nothing to see here. Past the amphipod. A little bit faster forward, please. That was a praying cephalophore, calicophoran. A little bit faster forward if possible, please. So we can see him coming. Center stage, coming up from below. That's a Tuscarorid radiolarian in the middle. So after doing this oik, thank you, to the right, there's a Tuscarorid radiolarian. So yeah, get a quick shot of this oikoplurid.
Well, yeah. More animals
Okay.
in there.
Thank you. And then on to the radiolarian to the right.
Okay, I heard you do.
This is a Tuscarorid. It's a colony with a glass latticework and a little protist embedded in it. There he is.
Down.
Yep, you want this, right?
Yes. Right there?
Yep.
Yeah. This is one of the Feodarians. Yeah, that's the colony. Oh, this is fantastic. Really intact. That would be fantastic. We're gonna have a triangular lattice.
Lo que estamos viendo ahora en pantalla es un radiolario. Así que alguien dice la palabra radiolario. Foraminífero Manu aparece corriendo dentro del estudio. Parece que es un organismo colonial. Se pueden ver todas las espinas alrededor de él.
I think the one with the suction is the one I have most confidence on.
Okay.
I mean,
La
we got
idea va a
nothing
ser
out of the big one.
intentar succionarlo para traerlo
This is
para la superficie.
phenomenal.
That's amazing.
And we'll be able to measure its cell density.
Completamente intacto, limpísimo.
John, we have a task ahead of us. Feodarian. Yeah, which have now been separated
It is a
from radiolarynx.
It is a Tuscarora.
But it is a Tuscarora, absolutely. I mean, absolutely.
So, do you think we've got enough video here? Can we
Ahí Cecilia
close the
comenta
iris
y
just a little bit
incrementa
more
la
so the
información
whiteness
que estamos
is
teniendo
a little bit
en vivo.
more,
Yeah,
less
see,
white?
the mesh is incredible.
No, I love how you
Yeah.
Incredible.
It's
It used to be
It's like a
regular.
loud.
Is it possible to shut the iris a little bit more?
You're welcome. Love
They're really beautiful.
I think we're up for the challenge.
There.
A little bit of purple on it.
See, that's the Tuscanarian, but with a much finer mesh. You know, the other thing that it always has two horns that I've seen.
Yeah.
Actually, two or four even.
What is it?
I think it's easy to say with these things.
One cell. One
Single
cell.
many cells.
Oh, I
Oh,
lied to
yeah,
you.
that. It does have a lat.
I lied to you.
Maybe it's finer than
Sí,
what
creo que la
they
idea va a ser succionarlo.
It's a colony. Yeah, it's a colony.
Okay.
I think, yeah, but the other horns are actually
that's cool.
coaxial to the surface, while normally they stick out.
Então isso é uma radiolária. Isso é uma colônia. Cada bolinha branca dessa é um indivíduo, né? Que tá, eles se conectam entre eles e formam essa pequena comunidade de vírus unicelulares.
Yep, put it in
A
7.
gente vai tentar coletar eles agora.
Dungeon divers.
Dungeons and Dragons dice. You know how they have those two-sided dice?
Yeah.
Because back then in NTSC video, you couldn't really resolve all the fine parts.
This
Yeah,
is
this I have.
Colleen Berkeley knows how to do these guys.
Yeah.
With the ROV device.
Tuscaroridae.
Thanks, Mother.
Ahora Manu les va a comentar un poco sobre los radiolarios.
RORID.
Welcome back. What we have on the screen is actually a Feodarian. A couple of us have been excited about catching Feodarians on this trip, so we're going to attempt to capture it. Now, these are single-cell protists that live in the deep ocean. We know nothing about their life cycle. What's very special is that they form these remarkable geometries, as you can see. That's a silicaceous geometry. If it's a Feodarian, it has a hollow silica skeleton, and you can see the periphery very fine zoomed in structure. You can see a triangular lattice associated with it. None of the rhizarians have ever been cultured. Oh, okay. It's about to go in. It's about to go, okay. And I just jinxed it. So nobody's ever been able to culture these organisms because we have no life cycle. We can't say how old they are. All we have are fossil records and some of the images from broken pieces. So it's very rare to catch alive. And on board we have lots of instruments, including confocal microscopes, to then image the cellular structures live. They are small enough that they require kind of 3D confocal microscopy to be able to image. One of the puzzles generally for rhizarians and radiolarians is what encodes the geometry in these cells. And this field is called biomineralization. As you know, lots of organisms have skeletons, and echinoderms have these mineralized structures. But for this case, it's a single cell that's encoding all these geometries, including triangular lattices. So if you were able to see on the top of that spherical surface was this very fine triangular lattice. So how does a genome encode these precise geometries using the cytoskeleton? We know very little about. And I think post studying it live, we'll probably plunge freeze it in liquid nitrogen, which we happen to have, and then we can process and do a process called FibSem to then really look at it very granular ways. Feodarians have a name that comes from phaedocyst, which again is sort of a waste pocket or the waste bag of a cell. And it's hard to see in this that you could see some punctase associated with it. And then this particular species is a tuscadarian because it happens to have these tusks. Usually they have 4 tusks coming out. This one only has 1, so it's not clear if this is a known species or a completely new species. They got separated from radiolarians recently, and they are now cercozoans. The other big question is, for most of these rhizarians, we also find them on the surface. Some of the surface radiolarians, for example, will keep symbionts, and when they go deep, they shed their symbionts, and so they potentially make migrations that go, 1,000 meters or 1 kilometer in the ocean. Feodarians don't make it up on the top, but other rhizarians like acantharians will be also on the surface. Woo! Ah, look at the bucket. That was tense. Oh, it's in. Awesome job. I think a first live Feodarian for us. Woo. And then maybe we'll share photos once we get this live in the lab and 3D scan it. It's in my bucket list.
Can I take a photo?
Yes, it makes me happy. Let me see if I can find a photo of a Tuscadarian. Yeah, John, John, did he leave? So we will need we should liquid nitrogen it post. Yeah. I think ideally we should do a pressure freeze, which we can. Good. No, we did some very large acantharians in Villefranche. Yeah. And then we got incredible FibSem data. I think it's just the bifurcation. So at the least, we can first get the structure out and then do extractions. I have a 4x, yeah, I can have a 1x actually, so we can do a confocal on this with a...
Picture of these from previous collections.
Yeah. You know, I have PDMPO, so we could let it grow silica, and that would be cool. No, it doesn't matter. We could know where the mineral would grow. I have PDMPO, I have calcine. It would be time. We have a few days. We just soak it in PDMPO for some time.
Does it have a membrane covering?
Yeah, the lattice is all inside, always. Yeah, yeah. Yeah. And then the silica is in these vesicles.
But, I mean,
And the
reason
vesicles.
it's in a sphere like that is for flotation and it changes in ions, or
None of this, like for rhizarians, geometry doesn't equate function as yet. There is nothing that we have geometries that look like a king's hat, or like imaginary, like Heckle was drawing them.
inside the lattice.
How do they offset the weight of the skeleton?
Why have a lattice? It must be using that volume for something.
Yeah, so they have vacuoles, massive vacuoles, and vacuoles are what are flotation devices for most of them. Because silica is much denser, but you notice it wasn't sinking. But in the lab, when we bring them up and they want to escape, we'll see them massively sink. But here, I mean, it wasn't sinking.
Is that another seducer? Looks seducer-like to me. Yep, it's a seducer. Omnivorous tunicate.
Whoa.
Nice shot.
Still descending. We still haven't hit our 1200. I thought we would be returning by now.
No.
Okay. That's good, that's good. There's too many good things to see.
1061.
Oh.
Jellyfish polyp. So that must be feeding. What's the point of swimming?
My friend Cecilia,
Uh-huh.
that
Is there?
is translating all the that she said.
The question is,
Oh, she's
Spanish, but
translating in
she-
Spanish.
She's
That is
translating
amazing.
the original chat is in Portuguese.
That is pretty amazing.
And she translated everything that you say.
Oh. Yeah, I think
sent
that's
her
kind of
photo
one of the
you.
thread.
But
That's
this is
one
guy
of the
that's
thread around
speaking.
the... Yeah, because even Spanish, Portuguese, like, yeah, there should be an auto, but it's not the same. When
No,
somebody
but
understands it,
No, but
yeah.
it's translating. But your English is really clear for us. It's easy to understand.
They could also float upside down and wait.
Yeah.
We have to get them some foldscopes.
No, I told her about the foldscope. She's googling them. She's going to ask.
Arctic Petriflora.
Ah, aquela medusa que tem coisa pra
Qué
cima.
buen helicóptero.
Tanaka Medusa.
we got any collectors?
We have one, don't we?
I think
Yeah,
we're all
but
full.
that one, we can't
D
rotate it.
sampler. We've got one D
We got
sampler.
one D sampler.
This looks kind of damaged. I'd rather wait.
Yeah, does it open it up? Another one?
I think it's dead.
So you can go left or right.
I have to give a quick shout out. Shout out to my friend Nicole from Ohio. Thanks for checking in.
Very healthy.
She texted you.
Once you put it back into the application, it's open again.
Do you guys want it?
No, he doesn't look healthy enough. Wait for another dive.
Die another day.
Oh, has he got more secondary tentacles than the other one? Oh, yeah, it's got 2 or 3.
I
If we can zoom in on him and count the secondary tentacles on the bell. Something weird's happening. Oh wow.
And net, everything comes damaged, unfortunately. This is the only way to collect these. So this is what we roughly collected right now. And in that ball we can't see, but it's mathematically it's perfect triangular lattice. And then there is a, like right there, there's a geometrical theorem that you can't have perfect lattices on curved surfaces for certain. That's what it is. That's what is hidden, which we didn't see. So the ball, but how does it know from here? You see that there's a defect here? Triangle, 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 but it needs to put a square to close the lattice. Like the same thing as on a soccer ball. You have pentagons and you must fit a hexagon on top to close it. The same idea applies here, but in that we know where to put it. This figures out through self-assembly where to put the square for this triangular lattice to fill, which is just crazy. It's
And now you have your own.
yeah, now we have to figure out how to image it. Yeah, but they are supposed to have 4, but we didn't have 4, it only had 1. So this is probably a different species. And it's not a Tuscadarian because that has 4 and not 1. Enjoy the broadcast and like swimming. It's just like we are all swimming in the ocean.
So far we haven't seen one of the paired ones, which is
But we made the call, we got the Tuscarora already.
Yeah,
that was good stuff.
We'll see the Philodendrons again. That was our first Tuscarora, right?
If we reach out.
I didn't think I'd see the day without this color.
That's our number.
another amplitude.
Can we go a little bit more forward, please?
It's incredible. I mean, think about it. The pressure down there, it's for a single cell, and they can handle. We are seeing them all the way to like 400, 300 meters. So every 10 meters is an atmosphere.
So
Around 100 atmosphere.
We're gonna,
Like crush anything. I don't know
then
how
we'll
many
fire
cars
another
that
500.
corresponds to. It's insane to think about.
Yeah.
Yeah, these small, very delicate things. I mean, it's tricky. It's isometric pressure. So it's pressure from all sides. So it's not our perception of how we think about pressure. But still, the fact that...
We're just punching down to 1200 now, are we?
Like the
Yeah,
range is so
of
large.
sorts.
If there's something really big, we're stopped.
We'll have to drive a little bit more forward to see something big though, because it'll just remain out of focus as it goes higher. Unless it's right in front of the camera now.
Yeah, but we will be coming back up.
And we'll report.
Yeah, but it's the same problem. On the way up, we've got to have forward motion too, where you
Yeah.
It's like,
need to find
Yeah.
part of
Yeah.
the, you know,
Oh.
what it is,
Maybe
and
more
try
versions
and
of
that's
Travel.
gravelly.
there's a lot of topics going on.
Take a look at the Down at the bottom. The head's on the bottom though. Okay.
Oh, it's going to infinity. See? It's like a lure line.
Look at the somatocyst.
This would be a fire sealer.
It's a calicophorium.
Attach to South Africa.
He says attached to something.
Yeah, who said that? That's funny. Oh, look at you.
It's very orange. So it's like when you zoom in.
Perfectly equidistant to each of the bones.
No, it's trying to see the somatocyst there, which is what we need to see.
She was listening. All the
Yeah,
information.
no, I think that's great. The pressure is a pretty Starlink's final force. That's one.
But I still haven't seen that somatocyst in the massive layer. It's not.
Yeah, right now,
because,
a big
it's
mystery
got that orange
in
one.
deep sea biology is this idea that is it possible molecularly to measure hydrostatic pressure? We know it's possible to measure forces. So like the last Nobel Prize on piezo, which is a ion channel, it can measure forces. That's how we can sense touch, and lots of things in the body naturally can sense forces. But pressure, we don't know if there is a molecule that can sense pressure. Because if you could, then you would know absolutely where you are, independent of light conditions. They would know. It's like having a barometer built in, but there's nothing that's ever been discovered that can directly measure pressure and would have such a large range. So kind of imagine just like light, like, you know, brightness and darkness, if there was an equivalent for pressure. And all of these organisms probably have them. Like if there is one common thing, would be that they have a sense of, because then in the absolute dark, after it's all dark, there is no information. So how do you know where you are? Then how do you even do a migration? Like what does it mean?
And do these organisms go to the surface?
Some of them, yeah, all of
But
them
they
have a
can-
distribution.
Yeah, but do they perform a vertical
We don't
migration?
know. The only thing we know, yeah, I think the way to think about pressure is, when we think about pressure, we have air cavities, and so in something like that, it's you know, the reason we can't dive is because the air cavity will collapse. But most of these organisms are all made with proteins and water, so we sense pressure by sensing the force, because the air cavity in our ear, in our lungs start to collapse and we can feel it. But for these organisms, it's different because it's all, there is no air cavity, but the pressure is extremely high. So at very high pressures, the molecules denature. So kind of like heat. Just like heat, if you heat a protein, it becomes like you cook an egg and it becomes white and denatures, the protein unfolds. So similarly with very high pressure, we know most proteins unfold. So you can, like microtubule is this one protein that makes rods, and they will unfold and destabilize. But not for these guys. Like even membranes, there's a beautiful paper on showing how membranes in some of the ctenophores, for deep sea ctenophores, are different than the ones that don't go that deep. They remain fluid under high
Very
pressure.
hard to make a living down here.
So there's a lot of,
Yeah.
Maybe she. That must be in Spanish.
yeah. Yeah, I think kind of I have some pressure chambers in the lab, so we could pressurize this. If we collect more of them, we could pressurize them and see. It's basically a little, just a metal box, so we can do some experiments. Yes.
Predatory events like sea feeding.
Yeah, so what they found recently for these jellies, they compared the membranes and the lipids.
Yeah.
Oh, qué lindo.
Yeah. Zoom in real quick. That is a copepod.
That is
Who
one
is that?
sexy looking copepod.
Okay.
That,
The radiolarian, and let's get that one.
no say that.
Oh, no, it's a shrimp.
The guy that is
Shrimp.
guiding the.
Yeah.
Yeah.
It's a
No, it's not.
I don't think it's a suggestion.
You don't think it's a suggestion?
No.
No, the antennae aren't like.
Oh, that's beautiful.
Yeah,
Tell some.
That's very deep. Very deep. Calinde means beautiful, right? Spanish. Calinde? No. Calinde
Qué lindo.
means beautiful.
Yes.
Calinde.
How beautiful.
Calinde.
Qué lindo.
Calinde.
Until it's lunchtime.
It's a question of Cecilia.
Yeah, I think so. A lot of stuff when we bring up does actually disintegrate. So many of these cells, some of them will disintegrate. Most of the fish come up really unhappy because they have these gas bladders and they explode. Oh, it folded them. Right? Yeah, it's folded now.
I bet it's never felt water current like that.
Renato told us that in this deep, most animals are red because red, you cannot see red.
Yeah,
It's
because there is,
invisible.
yeah.
Okay, he's excited now.
Yeah.
He's good.
Yeah, thanks, guys.
We ruined his.
Cool.
No, it would have been cooler if it had been.
Right. A lot of sensors on that head.
I just realized. So she works on fish. Yeah.
Yeah, she works with vertebrates.
I see.
Yeah.
She's a professor at the university that I study.
seguro es eso.
But I'm not prepared to say it's really more than 12 minutes. Just know that it's survive 12 minutes. The safety factor of 2 is in fact rated to 600 meters. The pressure tested is like 5,000. Yeah, it's fine.
Kind of hungry again.
Yes. At 11 we should
1,200 meters.
do it again.
Wow.
Deeper than this.
Are we gonna we're gonna niskening it?
Yeah.
All right. Niskening.
Shut a bottle.
You've
I told him to fire a bottle.
Is the bottles one and two?
Just one.
Just one.
Another one at 500
Somehow.
Yeah.
and another
Do a
one
sample.
at 50 on the surface.
Yeah.
No.
Okay.
No, I think that's a good idea. Why did it suddenly start moving?
Yeah, what's that?
Oh, I think it's the thrusters maybe.
Oh, there's one.
Fabulous.
That's the
That's
one.
it, yeah.
This one?
Yep.
Yep, so we want the next at 500,
Yep.
and then 250 and 2 at the surface. Or as close to the surface as possible. Oh.
Take us home.
Forward.
Up, forward. Whoa.
It didn't take us that long to get to 1,200. It took us
It's gotta be.
what it cost you.
Look at this one.
It's 9 hours.
We have an
A
artist
little under.
Oh, that's beautiful. I love the heteropod, the sea elephant.
It's hard going down, but
Yeah.
pushing back up.
You just keep sending Google out and get views. So what's current status? We have one B sampler left and we have a.
But the B samples are gonna leave.
Right, but in theory, if we get something that won't squeeze out, we've got
We have
one
one
B
B,
sampler.
we have one.
And we've got one suction for something that doesn't swim.
I don't think we got a suction.
We got a suction, but
Yeah.
it won't close.
It won't close. So it would
Ah,
have
okay.
to be something like
Something heavy.
heavy that we know is not gonna potentially swim.
Well, coelodendrons.
Yeah,
That's
some,
what
I was
I'm
about to
thinking.
say.
We could put one of those in the
one thing that's heavy is the rhizarian.
Sobre las preguntas ahí sobre la comida acá en el barco,
swimming with all
Pieces
the
of squid.
estábamos comiendo unos caramelos. En realidad acá en el control no se pueden comer muchas cosas, obviamente, porque hay mucho equipamiento. Y sobre el barco, sí, el barco tiene tiene un mini gimnasio. Hay 2 lugares donde se pueden hacer ejercicios, hay un mini gimnasio cerrado y en una de las terrazas también se puede hacer ejercicio. Hay una bicicleta fija y un remo. Y algunas cosas para poder hacer algo tipo pilates o cualquier tipo de gimnasia en el suelo.
No, I just said they were asking what snacks in the chat.
Who wants popcorn? I'll go make some popcorn. Anybody?
Dark chocolate.
Dark chocolate?
Huh?
Right?
One can never have too much chocolate or caffeine.
Oh, well, I was thinking of going up. There we go.
Actualmente nadie está con mate acá, no, en el barco. No trajimos el mate. Teníamos que traer tantas cosas que el mate no entró en la valija.
That's a good one. That's what we need.
To stay awake the whole night.
Okay, so let's start moseying up slowly. Try and keep some upward velocity and forward so the particles are coming at us on an angle. And so we can see stuff with a little bit of time to stop on.
Now that we're going up, I can build up
If you see anything you think is a big target, go ahead and go for it and save that delay in me telling you to stop so maybe we're not going to respond.
It's not. Oh, it's 10. Yeah, the last year was 3 hours long. They're now. They've been up since like.
In theory, we've got about 400 meters to cover an hour. A little less than that, but if you stop at what, about 100 meters for the boat?
About 125
Okay. All right, here we. That's a dead house. There's nothing in there. Keep going. I think it's actually like a dead jellyfish decay. It's just you can see it's not really good for boat structureless. House is kind of collapsing on themselves and turning into like a wad of white mucus when they're falling as a rule. They don't stay diffuse like that. Yeah, unfortunately, we have a schedule on the dives that we have to keep to so we can get the number of stations and be where we need to when.
Yeah, whatever you see, if you see something and they look to take it, if they're moving, or they'll done
Okay. Russ, do you want to, because we kind of look pretty well on the way down, what do you think about going up to the scattering layers there? Like popping
Well,
up
I'm trying to make sure we get enough velocity going up that we will get some time in the upper layers.
Yeah.
Which was a mistake last night as we spent too much time at depth.
So technically the ROV is without a pilot for a fraction of a second.
Yes.
Something we're after specifically.
Out of
So
pilot.
like speeding and stopping on deep star.
It freaks out for a second.
Yeah,
Back in
or
control.
Stygiomedusa we stop
Like-
for. Yeah.
There's no stopping
Yeah,
on
the
small
Star
things,
Wars.
right?
So we want the streaks coming at us obliquely. Perfect.
Looking for phantom jellies and big stuff. Sinking buses, yeah.
Good.
They're going for volume. Rosarium.
Yeah.
We have
There was
some.
a
Is there any reason for the suction to be over there?
suction shouldn't be on.
Well, it could be on the slope.
Opa, ¿qué hizo? Se fue. Salió. Chao.
Was that an arrow worm?
Yes, it was. Yes.
Yes.
There you go.
They have.
See, she's got it. She's an inverted expert now.
Yeah.
Nicely done, Mira.
There's a set of fins down the side of them that you don't normally see on the Keating Arrow. They actually do the back of an arrow.
He's getting going. I was thinking I was going the dark side getting into engineering. But I think it's the other way around.
Yeah, a little bit more, probably a little bit more velocity. That looks like a nice comfortable place.
And you went for the one that
seemed less like
fluffy. Oh, something big. Oh,
Something out
something
there.
big.
Here it comes.
What do we got? Oh. What?
Oh, it's another one of the big Mars?
It's
Yeah,
a big
giant
Fossil,
size.
yeah.
Oh, but there was
Oh, it's a arena with a house in front, that was where it was disappearing.
Oh, is that mucus off of it, Google? That little...
No, I don't think so. It's something else. Behind it looks like
This is a bison exobrantha.
But I'd love to scan this one.
Look at those tentacles. Look. Wow.
Do you know what kind of bison this is?
Yes, it's
He
Erena.
called the name of it.
E-R-E-N-N-A.
Oh yeah, that guy over here. Here's the larvation. Larvation left the house.
Saw that.
Nice eyes, nice eyes.
There's no house there. It's cool. I think it's really weird that the mucus is sitting right beside the colony.
It was, wasn't it?
That's trippy view.
Which Erena is this?
Charlie.
Yeah, zoom in. Nice structure.
Nice stick.
Wow. Looks like a shaggy dog.
Tudo tem forma diferente, né?
Sim, sim, sim.
É muito variado. Ai, tenho, desculpa.
Rub it on your face.
I wouldn't rub this on your face, it probably hurts.
Shepherd, look.
Não, senhor. Tem essa forma.
See, one of the dots you see along the Google.
I'd love a scan.
Yeah, you can do that.
Alright, want a DPIV, Joe? I'm most interested in that bottom section, the transparent area. The rest is super bright.
Yeah. Google, what are all these little circular spheres
If we
along
can
the stem?
also
That's like
zoom the science
gonads?
camera in a way that we can look at the laser sheet pretty well, because this one maybe might be a bit too big for my DPIV camera, and then we'll use the science camera.
This is big.
As the red laser light hits the tensilo, you may see very bright red spots on the tips. Oh, we're already shedding bracts.
Deu a contração dos dentócitos agora que chega
So it's
perto.
got to be 30 centimeters.
This is a fish eater. They've got really strong nematocysts. They're the bracts. They sit around the gonads to protect them.
Yeah, it might just wait a second until
We're
that
starting to
fluffy
get
bit gets
some
out of the way.
dissociation of the
Otherwise,
bracts.
that's just going to block all the laser light.
Oh,
Is that in frame or is it too far to the right?
There it comes into the frame.
Parece que pega fogo, né?
Turn on the lens. We can wait for the orientation to change so we can get the
Clear
experience.
part, like the markers and the clear part.
Yeah, the bottom part, not the fluffy part.
Is that a technical term?
It is a technical. That's why the scientific name is Fluffinosius rugosus.
Oh, right. Robustus.
Robustus, Fluffinosius softus. We had to make up some various classes of bony fish as a to fill a database without with some placeholders and all sorts of good bonians with skeletara, and all sorts of good bony fishy names.
Yes, it's the iris.
Yes, that's the iris. It's kind of embarrassing. It will automatically record and see something you can resolve.
You say Google this can catch a fish?
Yeah, it's a fish looks
I'll let it get real close.
Or is it so toxic it paralyzes?
It's, yeah, they're huge nematocysts. They probably be very toxic, I would say. And you have people
Getting
say
pretty close there. You're a little bit high.
deep channels can't see red. These have red for the rest of
there.
Until you check that. And other things. You're not just like, not just this.
bracts around here.
Thank you.
Yeah.
Just take one.
Well, we're throwing a lot of the bracts off. What's the strategy on throwing the bracts? Because they're the armor.
Exactly wrong orientation. Yeah, it's right behind the sheet. Just never should have threw out.
Hi, Mira.
Acho que andou batendo em alguma estrutura do
Martini
bolo
has kids?
e soltou
Oh, I
os
love
zoides aí.
it.
Olha lá, ó.
Ajá, salían todos.
Esse laser ele não é.
Maya,
Pode irritar, não?
can I see the babies one more time?
Not sure what I'll do with that data, but it looks cool.
Wow, amazing. The one from 2021.
Yeah, got enough.
But we also could only do a little section because it's too big.
Did you get enough scans of it?
So
Do we have enough
we're still waiting
scans?
for the right orientation.
Oh, really? It's have to be like.
You want to see the clear stuff clearly?
Yeah, the clear stuff is here. This is going great. The animal's like slowly rotating, so we can keep going on it. This looks like it's headed the right way. Yeah. Punch it through the laser. Through the laser light, I mean, not through the. Oh, yeah. Assist. It's you?
The very highly reflective thing.
Good. Great.
Yeah. Yeah. Cheat sheet. It's great.
Awesome.
That's good.
The best cameras.
So right now we're putting it through a laser sheet of light to try and do a reconstruction in 3D of the animal.
Okay, please.
And there's a camera that you can't see looking straight on at it.
Poor Joe, look at this.
Can I take a chair?
Has to look at it.
Let's
just turn everything off.
Não, ele. Isso, pulsa, só que eu não tô enxergando pulsar, eu tô mostrando o corpo da pulsa. Mas aí a outra parte é o
Keep the box and then.
que é onde tem os gastrozoides que é a
Use the
boca.
force.
Yes. Pure intuition.
E aí o outro
Essa cabeça é só propulsão.
só a propulsão.
Somehow that process, it always works.
I'd like to have a second dinner.
Me too. That's the chocolate. I'm gonna have third dinner.
Yeah.
Lasers already being blocked by the one side, the other. But then they just, that's why we're so weak. We're so transparent. Unless you want to apply regular light, I'll look at the laser.
It's crazy how it looks impressive.
On what one? Oh, really?
Falkor classic.
The same thing as you.
Keep going. Nice. All right, I think we've got it.
Okay.
Breathe.
C'est bon?
We
It's
collecting?
allowed now.
Are we collecting? No.
Turn the laser off so we're not tempted.
Are we taking it?
No.
No. Didn't think so. All right. Let's continue back along.
Pretty sure it's just
Heading up slowly.
Continue?
Yep.
Yep.
We don't see like no.
Yeah, it's kind of the city now, isn't it?
So one that got away lived to fight another day.
Yeah.
Oh, these are all the pieces, I think, are they? That we're going through still?
John, we also did Iris on it in 2021, right?
Maybe, yeah.
I feel like there's one of the few where we have both.
Was it one of the ones in the paper?
Yeah. Right now. We have
I get some international.
I think it's the one I tried to get the Xeno mark, and it's greater than 20. So I could see that it's greater than 20 something gigabases.
Which is a lot.
Yeah, and I put a lot of resources into it, so I got.
Oh, do they?
It's like 10 times.
They use your bank account.
10
They can
times.
deposit the money in your
Imagine.
bank.
Each cell it has 10 times the DNA.
All of those hang around like this.
Well, you know, there's this theory of constructive neutral evolution where you just get more and more complicated because there's more cost. So if they're just like kind of floating around and energy keeps falling on them, their genome gets more complicated, but it doesn't have any cost to them, so it just happens, it just keeps happening. They don't have any reason to.
Is there
There's
any
no pressure to
Because there's no evolutionary
Yeah,
pressure
you
to
also get.
reduce.
Is there any chance
We
that
used to, not anymore, but
over a
on
very
Skype,
long evolutionary
Skype,
history with lots of
spam
organic
people coming in on
events,
Skype meetings.
things like this may have.
Well, I was in on
The ones
Maybe just a touch brighter on the light or on the gain, whatever it is you're adjusting it with. That's too far now. We're never going to turn around. That was probably a.
Yeah.
That one put it in something. We're gonna get it. We've got to see it at the top in time to be able to stop.
I don't know that. I'm sure people have thought about something.
That's better. That gives me a little more time to be more oblique.
I asked for resources. Things like that.
Particularly in defense, there was like the grandma or someone there trying to join it, and then started walking through the house.
Is there like each zoid, each kind of zoid, the number of types of zoid and the number of replications of various gene clusters? Is there a correlation? Like, do they just have a complete set for one set of zoids and like, or is it? Because that would give you an amazing amount of being able to diversify.
It's like every time we see something, I just click.
Yes.
Yeah. And if it's go through one of these other processes, all right.
Yes. And also click.
And also click.
Because there's a symbol.
Do a little dance. It's a nice one. Look at the Millennium Falcon.
Or which one? Which one do you call them?
I think that's Superman, though.
It's interesting how oxygen concentration can go up. Yeah, but it's still, it was midwater.
A racing house.
41, 42. And then it's deeper going up, so they're going evenly, so they don't create, it's not being produced. So either the water mass comes in and has it, or something.
Get a little bit more upward velocity.
Besides very full, something about the light.
The focuses.
Something about this feels like I'm gonna find something.
It's the machines that aren't in
Oh, that's a house. Never mind. Keep going. That's another one of those mucus balls. It's not actually a house.
You just stay down.
Just stay down.
There. Halocreatid?
24 hours.
Or is that Arctopodema?
Oh, yes.
Arctopodema?
Oh, there's somebody fun in front too.
Okay. All right, up above it, there's something red. There we go.
Oh, Paraphilopsis. Again.
Paraphilopsis again. That's a small one.
Yeah. Does it have sets of 3 or sets of 5 tentacles?
Okay, so let's do a zoom in on this. Set yourself up.
That's such a pretty one.
Two, and 3. What? No, 4. 1, 2, 3. Yeah, maybe 5.
Acá el profesor Renato Nagata dice que es una sifozoa coronaria.
You want a closer look at it, or you want to move on, Dougal?
Zoom in on that, please. Yes.
Yeah, zoom right in on the bottom
There we go,
side.
There we go. 1, 2, 3. It's only 3. 1, 2, 3. So this could be a paraphilina. This do you reckon this would stand the D-sampler if it?
You know, it's pretty robust compared to some of the other jellies. It's probably about the right size for the D. It's going to be big.
Yeah. If we can get good morphology in this, and then we can get DNA on it, that would be pretty awesome.
Collecting?
Yes.
We're going right to collecting.
Yes.
Does- Okay, it is probably going to be about the size of a grapefruit, maybe a bit bigger.
You want to go on the D?
Yeah, let's go for that one last D sampler.
It's going to be about number 5, I think.
So it's actually probably not Paraphilopsis. I'm going to rename them now. I'm going to hire the first there. Skyfer Medusa. Medusa or Skyfer Medusa? I'm going to make it a paraphilina for now.
Yeah, is it a coordinate?
It's a coronate, yeah.
Yeah.
You have to look to see where the...
Well, it's smaller than I thought. It's not as big as a grapefruit. More like a tangerine size.
So you have to have a look at where the gastric cavities are compared to the mesenteries or the jelly that separates.
Oh, that's tiny. That is tiny for one of these.
Yeah, I would say he was small.
It's small, yeah.
The last one of these we got in the Gulf of Alaska, you'd have trouble fitting it in the sampler.
Yeah.
If we can get video like we did on the other one, that might go here, right? Just in case it's
Good job. It's nice and smooth.
But you have to look at where the statuses start and where the bits of jelly that separate the stomach pouch things.
Are you creating more slack as you come up
90 degrees to what
on the umbilical? Do you create more slack for yourself when you come up?
Yeah, that's why he's controlling the winch.
Yeah.
That's a bit too much.
Okay.
I can start to move the... Yeah, you go. All right. So,
Yeah, the other thing we'll want to do with the shilling is close those two tiers. Tops.
oh,
No,
the camera.
it's back in the chat. It's paraphililla, not paraphila. Or paraphilopsis, sorry, paraphilopsis.
Oh, it's not to see.
Paraphila is a different but also cone-shaped midwater jelly.
Yeah, paraphila has been done, especially
Thanks, Violet.
Thank you.
Oh, no.
Yeah, that's
Your arms pick me.
Yes.
Make a star out of me.
You focus a little bit through the acrylic bit.
Have to take it off of autofocus or it's going to keep screwed on the screws.
Oh, that's what I was doing. Yeah, that one is
Seems like it
Solito.
takes like a lot of time, years. I'm not sure whether small guys also.
Still on autofocus.
No, it's not out of focus. It's trying to.
Can you focus a little bit further? Oh, yeah, that's it. Keep going. It's going to hide just a second.
It's going to hide. Can we zoom back and move along?
Okay.
Okay. I'm trying to keep us on schedule.
Yeah. No dilly dally.
All right, moving.
So much time, so much plankton, so little time. That was a house. Just okay, keep going. Thank you. The lighting's better. I think it was a little radiolarian. It was formerly radiolarian up in the top.
Tem uma hora que tinha quase 200.
Or formerly foreign, as we were calling them last night.
Oh.
Yeah, sorry. The Rise area drive me crazy. All the traditional names are all completely trashed, and the relationships are so unclear.
Speak to family.
At every other protest meeting, someone is saying,
It's a nice bright
radiolaria don't exist.
Yeah. Yeah, we just need to get a symbol for them. It'll be like Prince, right?
Yeah.
The protist formerly known as.
Radio.
Yeah.
I'm getting there. Yep, I'm thinking about that soon too. I think it's 6 a.m. for the trawl, yeah.
Process these
Said she was gonna go.
Over here, another Arctopodema.
I'd say so, yeah.
Yeah. Is that right?
Yeah.
Okay, keep going.
Keep going.
Yep.
It's a Medusa, tracking Medusa.
Oh, there.
Oh, well, that was a Citadendron.
Do we want it, the last suction sample, or do we want a paired? We can always throw a second one in.
That one there.
The little thing that had the long arms, it just went back. I don't know if you can see it. It looked like a.
Yeah, it's right in the middle of the screen.
No, that's not it. It looked like drooping arms on a sphere.
Yeah, I think yeah.
It might be too far away, unless that's it right up there.
Yeah. I think you might have gone too close to
Yeah, maybe it's been blended.
Okay, we missed it. It's probably gone down the side already. Okay, a little bit more up velocity.
Was that just shimmering?
It's just water.
It was going to record in the spins.
That was, you probably got the comment on that, the Coelodendri. Yeah, pseudo. I can never remember the right end for it. I keep wanting to name it after the flower.
The Arctopodema. Oh, the Rise area?
Yeah.
Oh, yeah.
Okay, just passed through 1050 for meters. on our way up.
Okay.
Ooh.
Lick
Yeah,
Nagalma,
that,
maybe?
you want to have a swing back to that?
If we can, yeah.
Which site did you think it was? Well, there's a piece
I
of
think
it.
it might be Lick Nagalma.
I think there's still more of it, but we must have just blown it apart.
Yeah.
It's a little late to see it.
Yeah.
I think that was it, yeah. That was it.
Yeah.
The rest was just tentacles that are probably just retracted.
If it was Lick Nagalma, it has the tips of the tentilla have a vesicle with
Ooh. Another Persona?
No. Oh yes, yeah, you're right.
Milzai?
Soda Milzai, yeah.
Soda Mills.
Moving along.
Moving along.
Yep. We know what it is. We can keep going.
Yep.
Yeah. So, like many scientific names, that is named after an individual. Claudia Mills. Milzai is the second part of the binomial.
Tá falando com o pessoal daqui?
Claudia.
Sorry. Oh, did I get the wrong? Sorry, Claudia.
Soda Milzai.
Dá pra escutar.
Milzai.
Google
Crossota.
It starts with P, right?
Cross soda with a C.
It's a silent P.
Yeah. There's probably
It's so
almost
silent
every
you don't
letter
even
should have a
write it.
silent P at the front of it. Scientific names.
There you go. I like your description.
I think it's getting late and it's been a long day for the sound of the commentary.
Yeah. But you've got to be careful to apply that out too much. My children were terrorists, so if we put a T on the front.
So these guys have been at it too long. They're out of what they do.
Sounds like you guys
Down
are
here.
an hour 9
Is that- Nope, it's just a piece of bubble or something on the screen. So.
Never heard of the concept of an 8-hour day.
All the jellies useless sounds and they sit around. It's just another useless sound.
It's like 7 hours of productivity.
More vertical.
1 hour of paperwork.
hour to email.
But this is cool. We're there in the moment. We've got this new stuff going on the screen. It's like any minute it could be something no one has ever seen before.
Actually doing okay on time.
This is alive.
This is good.
And we still haven't seen France yet. It's good.
One day.
You don't have a payload that's kind of your average center descent rate on the displays. Just the you don't have a payload, a graph that shows your ascent or descent rate. It's just the line graph.
Maybe.
Okay.
Is that a squid?
Maybe.
No, it was a
No.
Tomopterid.
Really?
Yeah.
They probably normally they
Okay. No, I was just curious. I'm still trying to figure out what information's where.
Oh,
Oh.
something down. Yeah.
That the formerly
that?
formerly known as
Oh, that was
yeah, the one that he kept
Wasn't
telling
it the
us.
final assignment mob finally caught up to us?
It could have been.
Maybe.
right there.
Was it a
Formerly
Riseria?
Foran.
The one that
That's
we
what-
had accidentally called Foran, but I don't know.
Yeah.
It was,
Why don't we call it the sparkly Riseria?
yeah,
Just so
Call
we're
it
given
a-
a new functional name.
let's call it a Feodaria.
But then it could be anything. You need to
No,
it
we
needs
know.
a nickname.
Alusferia is
No.
the name that Hecker gave it. It's close, but it's not because morphology is so tightly correlated with these, and it is not the one Hecker saw. It's
All of these rosarians, when I first started out at the end of the 1990s, because we didn't know what they are, it was going NTSC, so really bad resolution. So it was like the dungeon die, the Daffy Duck head, and like they all had names, so we could call them names, always recording the same morphology in the database. But you read it now, it's like Daffy Duck.
Single, wasn't a partner.
No, I
It
don't
only
think
had
it
two.
was flexible.
Probably an
Right,
academic
it did
area
only
or
have two,
something else.
you're right.
They
It was
seemed
something
awfully
else.
flexible on the arms.
Yeah, they were too flexible to be.
Was it maybe a Sol Mandela?
I wasn't watching when it first came on. before it
Okay,
was
we missed it.
around.
Probably not going to
I'll
be
keep
there.
my eyes on the screen. Sorry.
I don't think we're gonna find it here.
I was
No.
thinking about Daffy Duck. Sorry.
Continue on.
Entire trade exchange. That's the other part.
Donald.
Oh, something up on top.
Small house.
The mass of mucus that used to be known as eudoxin.
Oh,
And Wicapura.
coming too fast.
Okay, we just passed through 1,000 meters.
Yeah, yeah.
On our way up.
Sense to get a little to the
Big thing, bottom left.
There.
Jellyfish.
Looks like a Helicreus minimum, I think. Let's just get a definite so we can put
Yeah,
it
I'd
in
say it's a
our
Helicrea.
habitat mapping data set.
There goes them.
All right. Forward on it.
Really?
Wait.
You saw that?
Yeah.
See how it looks like two bars with a
I
clear
do
bit
see
in the
what you're
middle?
talking about. Yeah.
Oh, it's got nice spines on the edges of the bell there. Just think how these little gelatinous things have structures like that, made of jelly. With a spine.
Creep it on it and
Now,
zoom.
if I was a Nakamedusa with a soft stomach
This-
wall, I would
this-
not be one
C'est
of these.
diferente,
This is the most spiky of the ones
Yeah.
we've seen.
As projeções.
This is the second morphotype. We had the tall, smooth,
Great zoom,
act
pilot.
dude. Yeah, nice.
Nice and smooth. Good.
I will take that as a cut.
I'd be much more willing to believe this helps you break out of the stomach of something than the knob on the Botryonema.
Still hasn't detected us.
And I've actually seen this in a Solnus' stomach as well. So they're definitely being eaten.
There. So where we're here,
They come out. Alright, Gibby.
Nice. It's a Helicreus minimum. Just look at that spiky hops. It's just out of this world. The other theory is
Okay,
it's for
nice
opening
footage.
cans when they get hungry.
ready to move on?
Yep, thank you. It's a can opener. The can opener jelly.
What's
It's a
the-
new-
what's
It has a
name.
common name, or are you making that up?
Trachymedusa. I've just given it a common name, and
Oh.
now that it's out there on YouTube and everywhere, forevermore it will be known as the can opener jelly.
I find it really hard to believe you are an influencer, Dougal.
I've just become an influencer. There you go. Okay, everyone, from now on in, this is the can opener jelly.
Oh,
No, it was a jelly?
We're good.
It's a rice
We're good.
area.
Okay, go.
I thought for a
Oh no, it's a little repellent nematid.
Yeah.
This is another one of these mucus sheets. It's not a house.
Only
Keep
at
going.
987. Just throwing it out there, team.
Throwing it out there, repellent nematid. That's a
Yep.
helicreatid.
Up we go.
Trachymedusa
We do
there
need to
for
collect
John.
water
That
at
little thing?
500.
That little thing, yep.
Alright, mine, I missed it.
Okay, keep in mind, collecting water at 500 meters. Is that an oik?
Right there. Like house.
It's weird in the middle there. There's two spots, not one.
Yep, like a pleura. There's no finger knobs or anything on the end of the
It looks very
filtered.
spiky.
No, it's just the normal corrugations along the side. Do you think you're seeing? Let's zoom in.
It's looking pretty corrugated.
Focus. No, yeah, you're right. It does look a little spikier, but it's
does, doesn't it?
smooth. It's from the right angle. It'll look like a pair of angel wings highlighting the animal. These are biblical animals. I don't see the animal though.
I don't see the animal.
Oh my gosh.
So this is a structure, we've heard to it as a house in this group of pelagic tunicates called larvaceans or appendicularians. It is a structure that's secreted on the outer part of the body of the animal and then inflated to provide an external food concentrating filter that the animal then sucks out a really concentrated food source from into its mouth and extracts the food in its pharynx.
Pretty cool. It looks
Okay.
more completed than the other guy. Is there an actual animal inside there?
There should be an animal hanging. It would be a little white dot you'd see at one end. It has a tail. It uses the tail to draw water in through some pre-filters that we're not seeing clearly on this house, and then push the water like a piston through that food concentrating filter. And then eventually the food concentrating filter is narrowed down and attached to the animal's lips. And that food concentrating filter then goes through a pharyngeal filter that pulls the food off and rolls it up into its esophagus.
Great detail.
That white spot in the center there. That's
No,
probably
that's
the
just
trunk.
probably a copepod or something. It's not the animal.
But the placement is.
The placement's
really
close,
to where you'd
but I'm not
expect it.
seeing a head on an animal.
Maybe found a nice pocket like here.
It must have been abandoned recently because it's still pretty inflated.
Maybe someone chomped his tail off and.
All right. Thank you.
That's what happened.
How can it secrete a house? There's a special series of genes that get turned off and on, and specialized pre-shaped cells on the face or the trunk of the animal that have this really complicated mechanism that turns secretory cells off and on, and then they blow it up like a balloon around the outside of the animal.
You should be able to, but I probably have to do some data cleaning first. And usually it's like one side yes and the other side no. You see this is a lot darker than the other side over here.
You can see the lattice panels on the other side. It's actually pretty dark.
Right there.
Kate.
Just a key? Okay, move on.
We're good.
Alright, I got it.
Chocolate's calling.
Yeah, this is the Uranus.
From today?
Okay, and a bit more up velocity.
That's from today?
Yeah, that's the Uranus skin a few minutes ago. Definitely it's a lot of cleaning to do still.
What is not what we want? Keep going. Sorry. No, keep going. I think. Oh wait, is that what we want? That is what we want.
No, that's something.
Is it? Yeah, it is. It's a bit damaged, but
Only at 974 meters.
it's very straight. They're not.
It's very straight. It's not weepy. And then behind it, you've got another jelly.
It's a repellent and matted behind it.
You get one shot at it, you either get. You want the rhizaria or not?
I think we're better off going with the one there were many of. Because
Okay,
it's
all right.
more obviously
Let's go.
more
Let's go through,
ecologically
forward a
important.
little bit more and have a look at this small jelly. Google, what do you want to put the jelly name down as?
Repellent nematidae. I don't know what it is. I'll assume it's a kind of cresota from the behavior.
Okay.
It's floating the same way as Milzai.
All right. Up we go.
It's another family that needs splitting.
What's higher than that?
Tracking Medusa. Oh, that's philodendron.
Yeah,
Oh, yeah,
that is
there
our
is.
target.
Yeah.
That's a proper one. And it's got 5, not 4, which is kind of weird.
6?
That's 6, my friends.
Oh, 6, sorry.
That is weird.
That is weird. Do we
Okay,
want a
this is
mutant,
going
or do
in
you
the
want
suction sampler,
the more common 4-legged ones?
No,
It's your call, John.
and this will be the last sample on the suction samplers.
You can put 2 of them in the same bottle.
Oh yeah, we could just collect them from now on.
Yeah. No problem.
And that means we are coming up with a full load.
Just imagine looking at this back in the days of the, what do you call it, round tubes, the cathode ray TVs. You'd be thinking, is this like a sea spider or is it a like what is this?
An awesome to know.
Yes, good.
I think it was born at the right time.
Yeah.
What's that?
Never used the 5 1/4 floppy disk.
No, the small ones I had.
3.5.
They weren't flop.
Yeah.
My first computer, we set it on the loading the software on the cassette tape and you walk away for half an hour and go eat your lunch. I had a full 64 kilobytes of memory. That's so silly, right?
Go.
But the science, you think of the early, say, 1900s, like all the subject around.
Come on, where are you?
On the beach, like, oh, no one's really looked into this. Look into this, and there you have, like, keystone species, papers, and stuff like that.
Yeah.
You feel like it was a little bit easier.
I lost
to
it in this
do
camera.
something. Subject.
There was so much more observance too. And now that we've got all the knowledge, like, we don't really look as hard before we kind of say, oh, it must be that, or oh, yeah, it's
a little jellyfish.
That, like, really looking
Oh yeah, there's a Bathychorus.
Yeah.
Okay, I guess we need to find another one.
Let's go to the Bathychorus and see how many secondary tentacles it has around the bell rim.
Yeah, that's a narcomedusa?
It's a narcomedusa.
Look at me. Whoops. Yeah, once it gets.
I'm gonna swing this way just a little bit.
I'm learning.
We're still after that silver end, but no.
I thought I might have seen it, but no. Okay. So Bathychorus is also known as the Darth Vader jellyfish.
No way.
Because the picture that got published of it during the Census of Marine Life looks like Darth Vader.
Okay, I'm gonna move on.
Helmet.
Helmet.
Yeah.
I thought it was because of the sound it makes. Suck it up.
Yeah. And Kevin Raskoff, who named it, hated it when we gave it that name.
Because it was whitish looking rather than black and therefore should have been a stormtrooper?
No, I think it's because he just isn't big on kind of common names. And it was the media kind of gave it the name as opposed to him.
Well, you're welcome
welcome to put the
No,
DPIV
keep going.
thing on the picture-in-picture if you want, in case the viewers at home want to see the 3D reconstruction.
Oh yeah.
Sorry? You're welcome to put the DPIV on the picture-in-picture in case the viewers at home want to see the
That's very nice for
reconstruction of the arena
so
from earlier.
that 3D reconstruction.
It's not very nice. It's awesome.
Okay, it's an awesome reconstruction.
Like, just did it right now. It's amazing.
Oh, there we go.
So
Over here.
This is the reconstruction of the arena we saw, what was it, 20 minutes ago? So it's a coarse first view of what we can do. We can see when we do a volume rendering. So this is not a full mesh model that we might use for, fluid dynamics calculations,
Yes.
etc.
Can you get closer to the microphone?
I can certainly get closer to this mic. So this is a volume rendering. That means we just use the raw data in 3D that we then visualize rather
Okay, I
than
think he's
really
gonna tell you
calculating
this
where the
is how
surfaces
it's
are of this
scared.
animal. If we do the latter, we can do fluid dynamics calculations on it, really measure the volumes inside each of the nectophores. So this is a first course look at what the data looks like, and we always start with this. You want to look at this jelly, right?
Yeah, this is the guy I think we got in Alaska with like, does it have 12 radial canals?
Caralho.
Let's count them. 10?
Ficou foda o bagulho, velho.
Is it 8?
Não entendi muito bem o que ele falou.
Ele tava explicando sobre o.
Just 8?
Tipo assim, sobre aí.
Or is this
O que,
another?
que
Maybe 8. It might be.
o instrumento lá
No,
faz,
the
né?
stomach is
O que,
so
que o scan
small.
faz,
Não,
quais as
Sim.
utilidades dele.
Não, isso
It's
aí
another
não
one of
entendi,
those
gente,
halfway in
que
between
É aquele.
Trachy reducey.
É.
Ahí
That's small,
just
that's
uno de los investigadores a bordo del
What
Falkor
name you want to
que
put on Google?
está encargado
Halscara.
de las imágenes
I
por
was
el
close.
escáner
Halscara.
estaba mostrando y contando una de las mejores imágenes que pudieron tomar de uno de los sifonóforos que vimos hoy, donde se pudo ver bien cómo escanearon cada uno de los pólipos. No pólipos, sifones. Cada uno de los individuos que terminan formando esta colonia. Entonces estábamos todos súper contentos que con todas las imágenes que fueron tomadas se pudo reconstruir súper bien la estructura de este sifonóforo.
Parece um negócio de CT.
Tipo uma tomografia.
A gente faz português. Só que sai muito mais poluído do que isso.
Sim.
Então, uma pós.
Shrimp? Or was it an amplified?
Juliana.
9:57. And we're not doing bad on time. We're a little behind where we need to be. What was our next bottle depth again? 500.
Take the U out of everything except glamour. Really great video about the introduction of the
Estas imágenes que se forman vendrían a ser comparables con una tomografía a ese nivel. Tomografía que nos hacemos cuando vamos al médico, bueno, ahora abajo del mar.
Something behind it in red.
Set up for the one first.
Look at that red thing.
I see the red thing, but let's creep in first of all on the one. Yes, focus.
What's this thing called? Sudusa? pelagic, carnivorous pelagic tunifish.
Which naturally is spelled
Yeah.
P-S-E-U-D-U-S-A. Of course.
So the name is a spin on the idea of Falk's Medusa. Pseudo means false.
But it could have been 4 letters
It
shorter
could have
and
been.
sounded the same.
Do you have it in here? It is actually a doliole. A doli.
Están diciendo que esto que estamos viendo ahora en pantalla es una sidusa. Parece que es un nidario carnívoro.
Just put tunicate other.
Yeah, so true.
Is this one bigger than the other one?
I think so.
We scanned yesterday?
Yesterday you scanned the dude that's on top of this guy. So there's a big one which we put into DNA, and then there's that little bug that you can see on the top. See this little dude near the stomach? So that came detached. You photograph that in your special camera.
Well, then I'd like to scan this with the laser. Shot.
Just.
We are light on time. So I see just
Vamos a
inside.
intentar escanearla para ver si podemos recrear la imagen.
It's not going to be 10 centimeters. It's not
No,
gonna
but
get-
the lasers are 10 centimeters apart. That's what those two beams are 10 centimeters apart.
The seducer will be probably smaller than that though, so you won't get both on the animal at the same time.
It looks bigger than it is basically.
Yeah.
Yeah, it's maybe
Pretty common in the
6,
midwater.
8 centimeters,
So
something
you
like
can
that.
do a scan size. There you have it. Can we do that? Another pass? Laser's already on. That's already in the frame. But if we could dim the RV lights when it goes through.
Ahí de nuevo se intenta que la medusa pase por el láser para pegar, para poder escanear diferentes partes.
I don't see it in the crossing camera. It might be high.
Así como lo ven en pantalla parece fácil, pero no, gente, es más difícil de lo que se ve.
I think it's close to like
Porque no siempre quedan bien las imágenes o no son muy claras.
Put a Z after it if you're writing UTC. Some detail.
which is how you abbreviate UTC.
Zulu number, yeah. It's an ISO standard.
Esse sistema em animal.
T as well.
Yes.
Ah, mas ele só vai pegar externo, né?
But now.
Vai
do
pegar
you
externo.
decimal seconds?
E esse aí tudo gelatina.
Nossa, você deve ser bem dentro.
How many? 3 or 6?
Claro, geralmente tem contraste também, algumas dessas coisas. Né?
Okay.
Was that a scan? Was it a roll?
Well, the lights weren't on. Oh, it's just outside of the. Oh, there it is. That's a nice shape. Yeah, there you go.
The money shot, the money shot, the money shot.
Yes. All right.
Okay, are we good?
Way to go. Yeah.
Oh,
Great.
It's a wrap.
joder, consiguieron
That was an efficient one.
Yeah.
Just do more.
100% eficiencia gracias a los pilotos del ROV.
Vai gravar
Easier when we don't get to do it for a half hour.
We are at 950 meters on our way up.
Oh, fish.
We're not going to hook on it, we're just
Ay,
going to try.
déjame ellos. Oh.
We've got fish people on board who've got no fish.
Ah,
Yeah, it was
Se
a
fue.
fast fish. little wee
And a conifer.
Canopy?
Yeah, it's okay. You can leave
Yep,
it.
keep going. Google's a professional. He can ID this on the floor.
And you can challenge me to say it's not.
No one in this room is going to be able to second guess you, Google. You're like scot-free on that.
But I'll listen if you can point out something.
It's with the sultry music.
Definitely.
Yeah.
It's part of the trend.
On this stuff, we'll believe you, but if
This
you're
is
going to try
wide
and
enough
sell
for
a
a sentence.
swampland in Florida, there's probably going to be a little bit more scrutiny.
I want to sell you a beachfront property in Alaska.
Yeah.
Yeah. Fairbanks, I was thinking.
You look at beachfront, well, okay, on the river, you get beachfront, but
Especially in
a
December.
little bit more vertical. Thank you.
Maybe it should be like a riverbank. Right?
God
Fairbanks.
knows. But also not in December.
Beachfront in Alaska doesn't really count for a lot when the water's only like 4 degrees C all year. It was an old house.
The Manatee Beaches. McKinney Farm.
And then when it's not covered in snow, there's grizzlies. And things.
Those are more forward.
Father-in-law's always talking about the time he's
Try and keep the particles coming out
newly
of
wed
the
couple
angle.
taking their pictures out there, stuck in the mud. It's not end well.
Much better thinking.
Hopefully, because the tide came up rather than the grizzlies came down.
I don't know which one I would have preferred.
They're going to have it in the end, right? No. Oh.
Voy a buscar mi teléfono, a ver en qué está mi carga. Voy a subir a buscar agua. Voy a ver la máquina de lavar.
Big.
Oh,
Size.
big size. Big size. Oh, look at the size of that. Is this a Stephanomia?
Sorry,
maybe?
Sarah Mander, your nap's over.
Okay, so the swimming end of this is going to be off
hope
to
you got
the
a
left.
solid 1 minute.
This is a siphonophore, right?
Yes,
This
it's
is
a
siphonophore.
siphonophore.
It's a Pythonec.
We zoom in on the tail and
Look at the
if
color.
The tips of the bracts have
Tenemos
got-
un
Yeah,
sifonóforo
oh,
en
yes, yeah.
pantalla.
This is Stephanomia amphitritus.
Very nice.
Va súper linda.
I want it.
All of it?
I don't actually want to collect it. I want to take another look. Look at that. Where's the rest?
Va.
There we go.
There we go.
Stunning.
Oh.
Oh yeah.
Junto
you
con
know
unos
what I'm gonna
pececitos de
ask
fondo.
Let me guess.
Tenemos pececitos de fondo.
This is nice because
Vamos a-
There you go. I brought backup, guys.
why am I not
Vamos a poder
surprised?
filmarla un poco más antes de escanearla y
I was
después,
getting looks.
con suerte, colectarla.
Yeah, this is a definite scan.
It's okay, lizard ghost. We don't know what that means. Sorry, Emily probably does.
She does.
Yes.
Sorry for saying.
You have to translate for us.
John was skibbity.
Some of those stomachs full.
Yeah, in this control room, you
That's right, I
speak
can see some
English,
eye spots
Brazilian,
in some of those.
Spanish, Dutch,
Are those
and
little
Sway.
little nests of nematocysts all along the outside?
Yeah, they've got pads
tiny
on the
little
mouths
ones?
on the ends of the
Yes.
bracts.
The head and some of the bracts.
Sí.
Please. Not collected, just scanned.
El ROV tiene acoplado una cámara que
Slight
filma en
orange
4K,
coloration
por
in the
eso
radial canal,
la definición
the ostial canal.
tan increíble. Incluso da para ver
Pads on either side.
los movimientos de cada uno de los individuos del sifonóforo. Wow. Ah.
So many mouths to feed.
Ah, qué imagen increíble.
Each one of them catching food, ingesting, sharing nutrients with the rest of the colony. All the sailors, which is like the larval breakfast attached earlier in life. So it would have been longer than just one stage. So, yes.
We're gonna have to rely on our auxiliary science camera.
Pretty big.
Marco, can we get the science camera so we can get on the laser sheet as well?
Oh, so good. Pretty good. Oh. So did you get everything? Yep. Metadata's all coming in okay. There's still like, formatting seems to change sometimes.
Okay, you get a maximum 5 minutes on
¿Mm?
the
Chata, llanta, ¿2? Ah, no, no es 11:30.
Yes, yeah.
Almost in sheet. It's starting to
You can
swim.
see it's coming through. And then every now and again there's a
Yeah, going.
What's the Google messages a second to see if that makes any sense.
okay.
How these are layered.
Yeah, the bracts are kind of.
Ah, me siento en una película de ciencia ficción.
Missed the top end. It's out of frame now, probably. Is that off frame? Leave the light.
I hate to say it, but it's almost a shame to have those lights off.
Yeah.
We need a multicolored laser so we can resolve color over the skin.
Yeah.
I've always wanted a
It's
brown
slightly
laser.
different.
They don't
I want
sell
a
them
white
really.
laser.
They don't really sell them. I want a brown laser.
Yeah. I did a rapidly revolving prism.
How far in front of the vehicle is the laser
20
sheet?
years ago. Because the slices weren't perfectly slices, they were slightly radiated.
Oh.
It's about half a meter.
I mean, got
Slices. Tem todos que são para propulsão e todos
It's just
que
a
são
bit
para
above.
alimentação.
I'd like to zoom in
I think
on
it's,
the
is it
tensilla
inside?
data.
It's inside, right?
The lights.
Oh, the nectosome's outside. Oh la la. Keep going, keep going.
Oh, close.
So close.
It was nice earlier when it was parallel to the sheet, and then in just like 1 second you've got the whole nectosome.
Can start from here too.
Yeah, that's
Lights.
Filma isso que eu tô sem telefone. Como se isso não fosse atravessado pelo cabo.
Pretty good. We got the top end.
That's pretty good.
Oh, it's rotated a little bit. This might be better.
Coming is coming.
Ah, qualquer que dê pra ver como tá sendo atravessado pelo laser.
Ooh, close. Second up to Rika. Can you need to do all in one pass, or can you add passes together?
We like to just do a single pass.
Is that in the camera now?
orientation the tail is going to fly.
Just a screen where you can see the Alan?
Or this one. It's there, but it's behind. It's kind of the worst orientation it could be in.
Right, yeah.
It's getting there.
The other thing is the very tip of the tail would be nice to look at. It looks like the larval bracts are already lost, but that gives us an idea of there's a few larval stages where we're not sure if it belongs to this or to Elastema foliacea.
The way that the joints on the cyphosome overlapped just looked a lot like Filigalma. Just like this really layered.
Yeah, they're very leaf-like and curved.
Just go around the whole thing. I'm just kind of obscuring the lower right.
To the right.
If we want to rely on the science camera, we just have to iris that down.
And he's pretty close to the beautiful.
Come on back.
Still feel him sticking out.
The little what, Sugi?
Back through. So close. There it comes. Lights. Maybe we should use the ram.
No, try this.
Oh, it's close.
Just doesn't fit.
It's too big.
So big. Or your cypher's too.
A little bit more to the right. You've got more transition.
We might have chunked him out. I know that
It's the flat.
it's a nice portion of cyphosome. That was a good one.
Test
I reckon that's probably
test
it.
the velocity correction. New software.
I just love watching the direction of the pilots and I'm just staring at a black screen with a few red dots.
thing we really need to see though is the attachment.
All right, we're at 10 minutes on this animal now.
The stem. There's a canal that is L-shaped or T-shaped, and because it's right up against the stem, unless they're detached, you can't really see it. And that's one of the giveaways, is whether it's this genus or another genus, which look, they're both really big and orange, and they've been confused.
Between this and Maris, or
this Halostemma foliacea, and they only were recently separated, like less than 10 years ago.
because the sativa is
Because they're so similar. Yeah, well, there are other things if you know what to look at, but that's the first thing you look at on detached necrophores, which you can't look at on this, so you've got to find other characters. But I think these, the pads on the end of the bracts are pretty useful for whole colonies.
It's swimming away to
Too many camera angles to look at.
So if you had a science camera, it's a little bit lower output. So if we're actually going to use that for some future animals, I'd have to find a way that we don't saturate it so much. Oh, look at that. Yeah.
Do we have a rat?
So once again, we need a wider field of view.
You need to what?
A wider field of view.
Yeah.
Lots of people talking about small animals, but always wider.
The red beam is trying to do a scan for reconstruction of the animal in a computer in 3D.
Optical section.
Are we looking Kikani?
Optical sectioning.
A good one, so we can head up to shallow waters to see what's happening at night.
That was amazing. Just
Nice. All right. Follow the route. Again, there's one.
one more.
Last time.
Easier if we just did it blindfold.
You need the lights.
I'm hoping that's okay. I'll do that.
Yeah.
They've got lots of DPRD up there.
They have. So the data with A.
I'd say maybe that was that.
See
We definitely don't have the char big enough for this.
the lat long is coming in
It'll
at
take a while to settle. Should we?
a quarter of a second, a fifth of a second between each reading. The depth, either really quick, sometimes up to 1 second delay. So it's pretty good. I think we're cool.
That makes sense. It's obviously coming from the Sprint navigation system. Because there's also USB out, which gives us less frequency. It's generally absolute.
Boink.
It's just too big to keep in the beam.
It would take too long to settle.
Yeah, I've been doing this for many years now.
All right.
It's weird. My brain kind of works on a.
When did you first go to sea?
I was working with plankton nets.
We're not collecting,
No, we're not collecting. We've got no place to put it.
Such a shame you have Monomia.
All right, let's move along.
Yeah, back then I could identify myctophids and like the fish and the shrimps and things by the light pattern and sometimes even just the way the face looked. But it's been so many years since I
No,
could
don't
fish.
hurt it.
It did swing through
I still
the
retain
iris
it
at one point.
sometimes,
Yeah,
but I
there's
can't.
stuff that
It'll
you
drift up.
can't get out of your brain.
Yeah.
I got loads of stuff from
Okay.
equipment that is totally obsolete name.
Yeah.
I can tell you facts about the old
Be free.
12S, the very, very first multi-beam assembly of ship. Totally obsolete.
Okay,
Sure it still exists.
up we go.
That's the thing with technology, isn't it? Taxonomy, I found, because even if it changes a little bit, you still need the original knowledge, so you can invest in having that in your memory. But
Okay, get the focus right.
a day,
Lighting right.
6 months on, and you may never
I just went by a big one of those mucus
We're
balls.
talking about computers. My first computer was something called Scan 2, 4K.
Need to figure out what to call those, but I'm convinced they're not old
The
houses.
same programs. It was designed for use with a paper tape reader. I didn't have one.
A little brighter.
So I just had to leave the computer on full
Okay, that works.
or manually type it all in again. Mind you, 4K there wasn't much to type in.
When you had an update, did you use a Commodore 64?
The second computer was a VIC-20,
A VIC-20.
which was the one that came before the
My friend had that, and they used to go to the program in machine language.
Yeah, I did a very, very little bit of assembly on that. But the first time I had to do serious 64-bit assembly was when I was in politics.
Just a touch slower.
I used a Rockwell AIM-65.
But-
It was 6502 development system. But I haven't really touched much assembly since then because I hated it.
I guess you got like a DOS 3 or something, and you're like, oh, wow.
Okay, a little more up.
Oh, great.
I remember that my first PC used DOS 3.3.
Great, thank you.
DOS 5 came out. And we all bought DOS 5 basically.
Too much up. Or more forward, whichever way you balance it.
3.3. And there's the word usage called networking stuff. But that computer got stolen when I was at sea.
Are the thrusters on this hydraulic or electric? Old house.
I think OS at least 7, maybe 6.
Oh, yeah.
880.
Job they use quite a lot of minutes. The old 68 times plate.
Before you could virtualize PCs, I used to carry 2 with me. I've got so many different Windows computers on my Mac, and then they went to Apple Silicon, and he said, I can't use any other anymore. Well, you can.
There you go. Yeah,
I've got that, and I've got more recent Windows installed on that.
Yeah.
Before because the ARM version will work, but the Intel version won't.
So there's not a version of QEMU for Apple Silicon yet?
I'm trying to think back. I don't think so.
That surprises me because there's certainly a version of Linux that runs on.
Is that a thermometer over on the side?
I thought it was a heating mat.
Okay, it might have been. It was just,
It's going pretty quickly with the M1.
could have been either without reviewing it.
And it just flies. I mean, those processors, because of the really tightly coupled memory and everything, really do very, very well. And in fact, Framework have done something very, very similar with their desktop machine. So there's no way you can upgrade them. You have to say, okay, how much RAM do you need before you want it to run? Very sure that's what you need. Or unless you just want to go for the maximum RAM price, just don't
Oh, that's one of the longest runs we've made without stopping. There's a siphonophore underneath us. But which I can't tell you.
This is January, I think.
Yeah, it's been broken since January or something. I've made multiple trips to that and have installed, but haven't been able to fix it.
How many got a mini do this for you?
No.
Sounds like a lot of wasted time for you.
It is, yeah, continually troubleshooting. I'm like, because I'm looking, everyone's got computers and I have to troubleshoot for all of them whenever I have a problem. Because we don't know infotech people. They just keep throwing security stuff at us and we don't have our way around. Whether it's firewall, whether it's Falkor crowd spread, whether it's whatever, verify.
Oh, you got
At 840 meters coming up.
I'm continually being locked down, but if you unregister and then register again, re-registration is fairly quick.
Yes, but we were assuming that our tourism would compromised and shut down or something. That's where if we lose access to Arctic for some reason.
Right.
Not sure what that was.
That's what I mean, a single point of view. Yeah.
Like the CrowdStrike stuff that happened with the
Fish, I think.
So you moved from Australia to Japan. Still in the middle of culture shock.
825 meters.
It's a kind of different photo. And that was back in the early 90s as well. It was good because Japan was still in the bubble economy, just coming off the end of it when it started. Obviously, it was still like everything was happening everywhere. And they were willing to give chances to, I was chief scientist when I was 28, I think. When I should already go.
That's some kind of a sparkly rise area, yeah. Small jelly.
But I was changing oil in my quiet focus and rush
1123.
stuff like that because there's no support system for scientists. So it's good for the hands-on learning stuff, but capacity is completely swamped pretty quickly.
So when you went over, did you know any Japanese or did you have to learn it as you got there?
I knew some. If people talked really slowly to me and changed their words, if I didn't understand, I could follow. But two Japanese people talked to each other, I could. But within a year, or within 6 months, I could do the spoken and hearing
800 meters coming up.
and the reading and writing was within a year. That was cold turkey for most people.
Couple of small jellies or something just went by lower frame.
Were they quite helpful in trying to help you understand what they're doing?
Yeah, well, I went just, I was enrolled in Bachelor of Arts, so it was a double degree, and so it was just learning Japanese and stuff.
house. Ooh, jelly.
It's a narco medusa.
I don't think it was a narco. I think it was one of the halocreatids.
Something down
Yeah, it's the hybrid dude that doesn't quite fit either. With the really long tentacles.
Ooh, what's that?
Whoa, is that a Bathus? No.
Soroi again.
Whoa, look at the size of those lobes.
I don't see the oracles yet.
Where are the tentacle bulbs? Can you see a gut? Oh, I see the bulbs. But I can't see a gut.
Okay, yeah.
You can straighten out a bit.
It's straightening itself out now.
Yeah, it's got no oracles.
There's no oracles. I'm not seeing them.
Alright, so it's an undescribed genus.
And I'm not seeing the kind of split off the radials.
Yeah, and the canals aren't golden. It's a kind of Illyria.
I can see the tentacle bulbs down at the bottom.
Is this something you can scan?
Yes. This is what scanning is made
It's
for.
big.
Yeah,
It's
it's too
tricky
big for
when
you,
it
maybe?
starts.
It'll be too big to get in the scan unless it's.
So?
It's huge, right?
That is quite big. Well, do it anyway.
Just try and get a lot of yeah, this video is great, the 4K stuff.
Almost looks like it's going to be very kind of Bolenopsis shaped when it straightens itself out.
You see the canals leaving the infundibulum and meeting the comoros partway along their length? So that's separates it from Bolenopsis. And it's got no oracles.
No, I didn't say it was Bolenopsis, but I'm going to say the lobes are coming up very
Yeah, yeah. I see
very
what you mean.
large and inflated in that
Yeah.
sense.
And it's got the projections
But
around the statist end that kind of.
And Bolenopsis would have oracles, so
We filled our descent, but didn't we? It's going to leak anyway. Oh man, that is scattering the light so much. So even though it looks
camera be centered a little bit more on the laser sheet.
even though it looks transparent to us in the regular camera, I think the laser line is scattering in the body.
Marco, you have iris or aperture control too, right? I do. If it looks really blown out in the red and we turn off the lights, maybe we can even stop
Look how
that down.
scattering that.
and there's our finally our Riseria coming down above it.
We do that again with fewer ROV lights.
My goodness. Awesome.
my DPUV
Okay.
camera is useless here, but the science camera seems to be capturing it.
The
Can you
science
zoom in a
camera,
bit
yeah.
more? You zoom on the laser sheet.
We get sizes only from the PIV.
Okay.
Yeah.
All right, we have an hour and a half left and we have 800 meters to
Can we
go.
put the lights back on and look with the regular camera?
Can we do?
Oh,
We're
you're
for one more
doing
pass,
another pass?
actually.
Okay.
Yeah. We just got the science camera zoomed a little bit.
Okay.
There's great. Oh yeah.
Oh yeah, that's nice.
Oh, that's great on the DPIV camera too.
Oh, that was a good one.
That was off frame though,
Push
wasn't it?
Straight back through for a bonus run. Yeah. Keep going. Yes.
Oh yeah, you got the right.
Was
the
that in
canals,
frame or
yeah.
off frame?
That was in the DPIV frame even. It was
Whoa.
the
That
only
was
way
good,
it could
yeah.
fit. Was
Yeah.
this orientation.
That was a good one.
All right.
All right,
wrap.
Later, Tina.
are we done with
Yep,
this?
we're done. Yep.
Okay. Nothing
We
to
got
see
no
here. Move
canister
along.
left, so we gotta do what we can do.
What am I going to label this for now?
It's Lobata
Lobata.
Lyria. Llyra.
Yes, brighten things up a bit. Thank you.
There's a whole heap of ones without oracles, and you know, we really don't know what goes where. But keep. Go check my copy process.
Right here. Patronema? Do I see a knob? Oh, it's Haloscaris with the spikes?
Did Dougal go to sleep?
Or is it
Maybe.
Haloscaris or Halocreus
Yeah.
with the knobs? That one's even more pointed.
Very pointy.
Wow. What are we labeling this, John, as?
The wizard's hat.
Lizard's hat?
The wizard's hat.
It's, I don't know.
Lizard's hat.
The lizard's hat, yes, certainly.
We need you
Some
to
kind
go.
of Medusa. I don't know.
Medusa. Let's start with that.
Yeah.
Halocreid.
Hallo.
It's a
Hallo-
halocreated.
Hallo-
That's too far. That's too far down for me.
Halocrea.
Hali, like an I.
Halocrea minimum?
No, it's a helicreate. I mean, it's that family. But
Okay.
Gotta go
That's it.
from there.
Helicrea today.
What's above that?
Helicrea today.
See that? Trade team
Like you want to be above that. It is a Trachemedusa.
Trust them.
Wait.
Yeah. Let's try and get a little bit of a zoom in on it once you got it under control.
How small is
10 milligrams.
milligrams? Is it something fluorescent or something?
No, it's powder. It's just powder.
Yeah, but that's what I'm saying.
It looks pretty white.
Maybe coming back.
I was thinking if you're making a solution, and then if that solution is fluorescent,
I've never heard that.
oh, something
Very
up on
nice.
top. Up on top. Right there.
Okay, and that
South.
was actually a dolly opposite.
Oh,
All right, that's it. Let's move along.
Go quickly eat. Oh, Google, are you here?
Google, people in the chat want to know how you make your coffee. Believe it or not.
Do you want coffee? Hey?
Because you're gonna make coffee?
You'll get
Oh,
coffee.
how I'm staying awake, is that the question?
Yeah. How you like your coffee is the question.
How do I like my coffee? No.
I think they want to come bring you some.
How does Google prepare it?
Right there.
Oh, something in the
Keep
middle.
going, keep going.
The,
There's a Larvacean
Pendicularia
house. Over here. That looked like a large Medusa. There we go. Another one of what we just looked at.
What do you found?
Halocreatid, some type, same as the last one.
Which one? Halocreatid minimum.
Minimum? Okay. All right.
The can opener jelly. Can I sit here and you sit there?
I have to go eat.
You gonna eat too?
Just give them something.
Okay.
All right.
We'll give them something.
Okay, I'll be
I come
Start
back,
heading
I
up.
eat,
Again?
I'll be back on screen. Okay, Dougal, where do I?
Trachymedusa?
Trachymedusa.
I'm not sure what those were.
Oh my
Scythe
goodness,
off to the
Philogama
side.
again. Oh, missed it. They had a skein.
You think it was an Algama, a fish?
Fish.
Oh.
Paralepid maybe? Oh yeah,
it was
we
a
are
busy depth.
cruising.
And that was a Solmissus, I think.
Oh my goodness, we're going so fast.
Well, it's a little fast, but if we
Yeah,
don't,
no, I understand.
Are we trying to hit
good.
the?
well, we're
getting
trying to
everything
cover the
up.
rest of the water column in the time we've got left, which
Oh,
is
I see.
just over an hour and 20 minutes or so, right?
Oh, those
There.
are
That was a repellent.
Yeah.
Medusa.
Yep.
Hello.
Yeah, Trachymedusa.
Trachymedusa. Repello.
Repellent nematidae.
730 meters.
There's a lot of uncaptured diversity in that family. But it's true of almost all of them. so-
Uncaptured diversity. Yes. Once we can name every one of these dots, I would be happy.
Is that a fish over on the right?
Left.
There's another one of the.
Medusa.
I thought it was a Rhizaria. Yeah.
On the left it was.
Oh, I'm
yeah.
not paying attention. What am I doing? Did we catch the cylinder?
But we did get your Tuscarora.
Yeah, I'm happy. Trust me.
Philodendron next time.
Yeah,
They haven't seen a lot this dive compared to the other two.
Yeah, that's right.
Small something.
And also the one that I.
And you were after them, I think.
3, but now 4.
It's time for caffeine hit.
But when Dougal said no, we'll get another one later, he promised $100 if you didn't get one.
I heard that part.
Yeah, you heard that too? Yeah,
I heard
he does remember saying it, but I heard it.
Pesos.
Yeah.
It's 100. It's 100 Australian, so it won't go as far.
100 Australians.
Or
Yeah, let's-
worse, if it's 100 yen, you're
$20
really
US.
getting a lousy bargain.
Let's settle.
Yeah, let's settle on a currency.
Oh, what was that, Dougal?
Mine.
It, mud. I would have said it was a collapsed house. So
Same thing.
Oh, on the left.
yeah, that couldn't. We're going too fast. Idea.
Oh,
Here.
yeah.
What was that? You
Oh.
got him? Yep. Is it a fish? Is it a plane?
Some kind of a jelly out
Super
there.
jelly.
No.
I haven't seen any turtles yet in this dive.
It's turtles, little sperm whales, right?
Oh, please be a Rhizaria.
No, it's a keating.
No, it's a Keatingnath. Oh, lunch bag letdown. Okay, continue on.
I'm supposed
Is
to
it
be
really a
good.
keating? Yes, it is.
No, you take that.
That's a keating.
You
are?
Do
No, I
once
think
I
I'm-
eat.
think I'm here until the end of
Okay.
the night.
Super exciting.
What?
Super exciting.
It worked? What?
Turmeric works.
I knew it!
Yes. It's the most gorgeous image.
This is amazing.
I mean, it's just fantastic. I'm
What
just
works?
getting too excited.
Turmeric
The
as a
chef-
fluorescent stain.
We are using turmeric to build 3D
Turmeric?
models of
Yeah,
hybrids.
yeah.
Yes.
You're cooking up a nice
A
experiment
nice
there.
curry.
Haven't you?
I talked to the chef and he was like, "What the hell are you on my turmeric for?" And the
And you
images
gave him
look
like a kilogram.
just gorgeous. Yeah, he gave me a kilogram.
You just look good and you just see like a baked fish.
Yeah. I mean,
That was something.
absolutely gorgeous. I'm getting muscles. I'm getting like the material. This is the early scan
like
that
my
I
mouth.
did
Oh,
with
okay.
a massive. But once I changed the context,
That's so
and then
exciting.
we discovered with that
Yeah.
that the hybrid eyes have a hole. You don't see this cavity, but the eye
It's so
covers
sticky.
out.
700
and
meters.
then it goes under.
Slow down just
No,
a
Oh, there's
touch.
the radio. There's the.
There.
Yeah.
We're gonna be able to go back, forward.
Yeah. On the right.
I think we went
That's
past it
amazing.
though.
I'm really happy about that.
I am jumping up and down. I just was with Jan and we now made a plan to like scan 30, 40 of them because every one of them we don't have the geometry for. And then we found some baby hybrids
Oh.
riding on a mama hybrid.
I think we should keep going.
Labeled that too. Okay.
Okay. Okay.
I'll be back.
Okay. That's awesome.
Just down there it is, right over there. Oh, is it another one of the rhizarias? All
Oh.
shiny?
Oh.
There we go.
Oh yeah,
Yeah,
yeah, yeah.
yeah.
Oh.
Oh yeah.
Wow.
Is there any chance to collect this?
We've
Yes.
got no buckets left,
It is
sorry man.
not
I
a
thought we
forearm.
do.
It's
Yeah, it's
Ferodarian.
it's the rhizarian who used to be known as forearm.
No. We're gonna,
Yes.
we're gonna
This
change
again.
this taxa. But
Decide
only if
quickly,
can
we have
collect
one
it.
suction sampler and it may not stay in.
Yeah.
Yeah, let's suck this one.
We can't. We don't have any
No,
substance.
but in the same bottle.
Yeah.
There's one
With the
bottle, but
open
we
one.
can't move
This
it.
is not
We
gonna
can't
swim
shift
away.
it out of position.
So it might.
It's
Fly
not gonna
out.
swim
It may
away.
come out.
What? Okay. It's the one that might. Yeah, that's okay.
It's all right.
It might be caught in the shadowgraph tank as well
It will,
because
yeah.
the pipe goes up
Yeah.
on both ends.
Then
Yeah, we can do that. We can do that.
Wow.
We get to change what.
It's
people
actually
think
one of the
this is.
biggest ones we've seen based on
Yeah,
the
let's do it.
cameras.
Let's take him.
I mean, N of 1 is hard to prove. N of 2.
Still not statistically significant. We're gonna have to get you another 3 or
No,
4.
No, the same animal.
Let me know when you want that ran up there.
What if you?
Are we gonna finish something?
Yeah, suck it up.
Yeah. You can,
What if we give you the same animal 3 times and tell you it's something different? Does that work?
For taxa, as long as it's that
Look at
same
it from
thing.
3 different directions.
Yeah,
Yes,
yeah, yeah.
then
So why are
switch to
the
size of 3.
so why are the silica arms so flexible?
No, they all feed.
Yeah.
Glass,
You-
if you look
you-
at the elastic modulus
Yeah,
of
it's
glass,
pretty-
It's like fiber
flexible.
optics. Fiber
Yeah.
optics
Oh, it's like that.
surprisingly
Yeah, that's right.
flexible.
It's like that kind of.
it's the elasticity
Yeah,
goes
yeah, yeah.
T to the cube
Yeah.
with thickness.
So what
Yeah.
you go up to this guy and then
It's
you
very-
take your pigtail of
Yeah.
your hair and you like entwine it in this and then you can talk to it. Like,
Yeah.
Yeah.
Maybe
That's Avatar,
there's the
right?
potential here for a synthetic fiber optics that would work faster and better. There's
That
big
is-
money here. If we could do that,
So what's not visible
It's
in
so
that
crazy.
photo is that the
Yeah.
tips, it bifurcates into thousands of more fibers. So this is turtles all the way down. Speaking of turtles.
So it's
It has a
flexible because it's really
disc roll.
thin, and
That's it.
It's almost
Elasticity,
like
yeah, and it's hollow and
yeah,
has silica.
Yeah.
And then what we got last one was really mushed up.
This is amazing. Yeah, I guess
No, I
that's
mean,
why
it's
people thought it was like.
Yeah, 4M people thought
organic.
because it's my
Yeah.
tubule, but that
Yeah.
one makes
No.
sense for this lens scale.
Yeah.
And then again, you know,
That's what-
in the density, you don't see the. I mean, it doesn't have the shell.
No.
It has phaedocysts.
Yeah. Oh.
And it's all labeled as 4M.
I can't see a brown body that's not in the center though.
I'll show you in the scope.
So
That's
close.
multiple bodies.
Yeah.
Yeah.
Yeah, I have phaedocysts.
Not a
in there.
single one.
No, not a single one. Multiple. I mean, here you can't see that because the.
Oh, is that a Piobius?
Really?
We might have seen our first Piobius just then. I think.
Outside.
Coming off the side, the right-hand side it was.
You see it in there that.
It's not in the frame grab now. I'm getting it so that we can find it again.
No, up there you mean? That's not could be a Piobius.
No, that's
It's
a
a siphon.
siphonophore. The one I saw was down here. Don't tell me I don't have Piobius in here.
Down the tube.
Nice.
Okay, keep an eye on the suction sampler.
See the tube? It's sample.
So we have two.
Okay, good. Shut down the pump.
Both of them.
Oh, where there's one Piobius, there's more
Oh,
Piobius.
okay, perfect. It got.
One thing
All right, let's hope we don't lose it.
Which suction sampler
We
canister is that?
can't move it. It's just going to have to risk that it doesn't get blown out.
I gotta write that down.
Yeah,
That's it. So you know, sample. Biology sample.
Okay, we are at 694 meters. We are boogieing our way to the surface.
John, where did we get the?
Is that a fish?
Area? Which sampler was it in? It was also in 6.
What? What other one?
The Tuscadarian.
Oh, I don't remember. Yeah.
Tuscadarian.
It's in the database here, but I didn't write it on paper or anything.
Because it's not listed here.
Wait a second, let me.
Yeah, Feodarian. No, it's there.
Let me.
Success.
Does it get a
Do you want me to poll the database?
Just
You
so
okay?
we know
How do we do
which
a sample
sample. Was it on 7
Oh, it comes
ID?
from Just wait a little while. It takes 60 seconds to run up.
Okay. It's listed. So can you list that as a
I got it.
unknown Feodarian
25. Yeah.
as the sampler 6?
You need some tag that lets you distinguish between the different unknown Feodarians that we're probably.
You want
Rather
to
than
be more
Nematidae.
descriptive than that.
Yes.
Yes.
Up, little Medusae,
oh.
Arctopodema.
Other Nematidae.
Something.
Oiko.
Something. Yeah, like house probably.
I think it must be sampler 7, no? Because
We're looking for.
what do you use for your reconstructions?
The Tuscarorid is.
Reconstructions? What do you use usually?
Combination. I have a.
SS7.
Sort of routine
Did you get that, John?
stabilize things and then
Tuscarorid is in
House.
suction sampler
Okay.
7.
frame stacks that are stabilized.
Tuscarora, I got it. Yeah, it's there.
Yep.
Using the crossing cam.
Any other ones you needed to know?
But
No,
not
it was already
for-
written
so
down.
There's
That's
really
it?
any
Yeah.
information there to use. 'Cause
Pulsing
we need
data
relative
task.
positioning and there's
Water
no
column has
fixed.
Back to capturing.
That's why I was saying that flow is a very important.
Oik.
At least in the steady state, but I guess that's not steady. But then after cleaning the data, process it.
And just so there's slicer.
I'll skip the midnight.
3D slicer to make the 3D models, and honestly, lately that's where our pipeline has stopped. So we can make all sorts of measurements on that for measuring the internal volumes and stuff like that.
So you don't use MRS.
Propellant and matted.
Yeah, the problem with
Something else in the foreground that I didn't
Maybe a
quite catch.
house.
In some sense, the next generation tools will be much better.
Yeah, I think the big problem is still the Iris stuff, finding good reference frames to. So how do you
Thank you, that's
describe this
better.
flow field relative to this other?
Small jelly down there, we're not stopping on it.
Halicreated, I think.
Reference. So just defining that clearly is not so easy when everything's rushing around.
Another
It's so much
house,
easier
the
watching
jelly.
this than the YouTube video. Like it's not as blocky. There's no lag. Like,
Fish. Oh, there's siphonophore down.
Niffies.
When it's these, the fish.
And all they've got is this lager.
Yeah, well, no, I mean, it's good. I've done a lot of good stuff with that YouTube thing, but I mean, this is amazing.
Do we get it or do we go
It's only
past
fair.
it?
It's like your experience with the NTSC cameras. That's what people get.
Yeah.
Can graduate. Like, and then in a few years, we're just looking at 12K cameras, and then they've got 4K.
Yeah, we may have just waited, not been able to stop in time.
Was that a slow Mandela as well? Man, thick and fast.
Okay.
All these targets.
It's a washing machine.
It was some kind of a Feiseneck that we
We're heading
weren't
shallow
able
to
to
see
stop.
where all the action is, but all the action's already here, man. There'll be even more.
It
when we
looked
get
a lot
shallow.
like the White Mars,
Yeah?
but we weren't able to stop on it. Like it was a long cyphosome white.
Orange gastrozoas?
No,
No?
I couldn't get any color off the rest of it. It was too far away.
Hmm. When I think about processing the siphonophores that we've already caught, I have an allergic reaction to catching any more or looking at any more. And they bring on the ctenophores.
Are we just on like day 4 of 10?
Where'd you get it, on your hand?
Oh, yeah, I was stung. It was like it hurt quite a lot, actually.
On the hand?
On the hand, yeah. And then trying to rub it off and then going, ow, that just stung me again.
Still rotating?
If you rub it off, there's a trick. You gotta, like, your eyeball neutralizes it.
Yeah, I tried to like peel back my eyelid.
Some kind of a
that I
blown
could
away
rub it off
scythe?
with the inside of my eyelid. But another classified.
Where are your turns in the tether? Or you never end up with that issue.
Yeah. See the T.
T? Okay.
Like going
Well, yes, it sometimes does move vertical.
We're untwisting, are we?
Try and get
Something
our
big at the
nose
bottom.
pointed back into it.
I
White.
got my blinkers on, don't worry.
You don't.
Wake. Okay, back in business.
Ah, it's just a house. Dang. Okay.
House in the middle of the stream.
Oh, house. Boy, house.
In the middle of the stream.
640 meters. Small site for something over on the left. Or sorry, on the right. My other left.
Are we calling that, we're calling that risaria that used to be a forearm a squiggle now, are we? All right.
House.
Oik.
Another house. Another house.
And that's a repellent nematode.
Siphy.
Did we just toss that away? Yeah.
Yeah.
That was a small scythe.
What were you? You looked like a Ficenex.
What type of scythe do you want it called?
It's a Ficenex.
I think it was an Algama?
I think it was a Frillagalma, but, you know, Ficenex as far as I go without rewinding and looking at it frame by frame.
Oh,
So,
Missus.
Mrs. Thank you very much, Joel.
And another sparkly. There's another house. Stringy thing.
It's all misses.
House. Fish. Not sure. Sparkly. House. Oh, bog. Something. $615 on our way up, about an hour left in the dive.
I think at the speed we'd come across a squid who hadn't seen us
House.
yet, wouldn't you? House.
We got a lot of ground to cover still, 60 minutes. We got to come, Somissus
Ah, missed it.
and another small jelly of some
We
type.
got a budget of 10-minute stop for Stygia Medusa.
Yeah. Although I prefer a deep star reticulum, actually.
600 meters. At 500 meters, we have a bottle
Deep
of
Star
trip.
Reticulata?
Help me remember that.
I only know Deep Star Enigmata.
House.
Enigmata, there's a reticulata or reticulum or something as well. A reddish one. I've only got the other one in my database right now.
And a barrel. Natural.
Got quite a few Stygia Medusa because we caught them down in Antarctica. I dissected out the embryos. They're pink when they're babies.
Everything's full.
Coelodendron.
Too late.
Good night. See
Good
ya.
night.
Thank you.
Got it.
House.
Oh, there's some new targets going past.
Probably a house.
Another rosarian.
$581 on our way up.
This is what it feels like to be vertically migrating.
Rise, rise area and sun type.
Turbojet.
Yeah.
They're Colubanema on steroids.
I feel like I'm a pyrosome right now. It's kind of shooting along. None of this pulsing and like changing the speed you're encountering the particles. You're just like, meh. I feel like I'm one of Bruce's AUVs.
Was that a scythe over on the side? They were kind of stringy,
Yeah, I think so.
still diffused with a lot of mucus out.
Mm.
Yeah, keep going.
Coming up to 500.
Oh, yeah, coming up to 500.
500, we're doing a water sample, right?
We do a water sample at 5. And there was a house just went down the side, I think. Small
Helicopter
scythe, or is
tail.
that a?
Praying tail.
Yeah.
Oh, is that a fish?
Still forward. See if I can find the inside.
House over here, a squid over to the side.
Yeah.
Let's go take a look.
Ghost, am I leaving you?
Hangs around.
There's food.
Right down
There you
there. Is that finally a
Squid.
cockatoo? Or a standard squid? Standard squid.
Looks like an
Standard
almost
squid.
strethid, maybe?
Plain squid.
There's two, because there's one off to the
Vanilla
left
squid.
too.
Pretty fast.
Going down.
Why is he going down? What's down there, dude? You should
I
be
think
up.
he's gonna
Not
just
us.
make us
Shallow.
race. He's not
Food's
gonna let
up
us
there,
get any
man.
closer. Oh, there was
it's
another
Sedusa,
seducer.
yeah.
Who is this
Oh,
squid?
it is. Nice.
Whoa.
We got him tired.
Where's
he slowed
Heather?
down.
Where's Heather? Yeah.
All right. There was actually another one hanging out in the area too. Saw it in the background while we were chasing this guy. Whoa,
Okay.
there we go.
All right.
And there's a Tomoptera, yep.
All right. Back
right.
to ascent.
Yeah, we can't hang
Nice
around.
squid.
There's too many things.
All right, sorry. I saw him. I couldn't go without saying, "What's that?"
I missed the Seducer.
How do
the
pyrosomes
validation
move?
too.
Pyrosomes are a series of colonies with their exhaust siphons all pointing toward the middle, the whole their lumen, and they use cilia to propel the water through them, and then that provides an overall kind of jet propulsion to the colony with the water being pushed out the atrial siphon.
I'm gonna close my eyes. Otherwise, we're just gonna keep seeing things.
Yeah, but you at least can annotate. That has value.
You can too. It's all in the
If you
state
like
of mind.
annotations on, at the granularity of squid,
Yeah.
worm,
Yeah,
fish.
because we can get back to them later. Everything like that's
And
useful.
a something. Never mind.
That was a decent sized squid.
Bibliotone.
Nice.
Was ist das?
Okay.
It's a Tomopterid.
Hey Russ,
Yep.
we're gonna attempt to close those slots before we head up any higher. while it's
Okay.
all-
Yeah, you good with the manipulator?
Yeah.
Okay. Good.
We are not fighting them.
Yeah, we noticed them mid-dive and we weren't sure what could be done about them.
Fine, just hold your position on the screen.
So, Mrs.
Yeah, I was trying to get in on the action.
I know.
He's like, let me in that D sampler.
Yeah, give them good.
Gentle touch.
It's a bit dangerous.
Oh wow, okay, we're gonna try.
Seal it
Oh,
in
there we go.
There it goes.
Console.
Photo
You got that
bomb.
one?
You get it, Michael. So
Yeah.
I'll put it, but
Wow,
all
Ben.
Kilo,
Nice,
Chaz,
you got
whatever.
Oh, wow.
Jeez.
Nice one. No pressure.
Wow, Ben.
Oh, yeah.
Nice touch.
Show off.
Well done.
There's no fish here for him to catch in a little butterfly net. He's making his own fun.
Okay, which Medusa was that? I think it was
50
a
meters to water
Helicarrier of some
collection.
type.
50 meters to water collection. Another highlight coming up. We're gonna filter.
It was a big day today.
It's still a big day today. After they come up, we're processing them.
Really interested.
Then we're doing a troll.
Yeah, so when are you sleeping?
Sleep. Sleep. Yes. Yes. Third of May, I believe, was my next schedule for that.
Jeez.
What day do you fly out?
3rd of May. And then the 1st, I think. The 1st. And I lose a day, I think. I get back on the 3rd at night, and then I'm driving early the next morning on the 4th.
Flying first class, I see.
Oh, yeah. First class, yeah.
Yeah, always.
Right up near the back.
Right by the bathroom.
Right at the back next to the bathroom. In the middle seat where you can't recline. Yeah.
Stopping in 40 meters.
The way you
to
guys are
triple
laughing makes me think that's where you're normally stuck as well.
I think we all are.
Nothing but the best for us.
Because we make tons of money.
It's to get us used to diving in submersibles, right?
Yes.
You like
No
that
space.
cramped feel, you know.
Yeah.
Which one do you go on?
Is that a submersible window or a tiny little monitor?
Yep.
Hey, the Shinkai.
Me?
Oh, you?
No, I haven't.
You haven't been on a submersible?
No, you guys. This is.
Come
I don't know
on.
how this has happened.
I've never been.
You need
If
to.
somebody wants
Okay, let's
to
You need to make
invite
some
me,
tech that
I
only-
will. I even did some engineering on Alvin, and I never got to go.
Never, huh?
So no, I know, never.
You need to make one of those things like the jet fighter pilots have, where they can just look at an animal and the electric pan tilt will like put the camera there and focus it as well. But
Our
then you
ML
I do see
tracking
a
does
sun type.
that.
Yeah.
We have that.
Heating,
And actually, when
But
we're
it needs to-
automating tracking on the vehicle, we don't have to adjust the focus at all because it keeps the relative distance constant throughout the track. It's really nice. So maybe that's the next time we come back, we could use it as a pilot aid. So when we're coming on animals, we could just click a button
house,
and automate tracking.
other house.
If you got a camera that could do that, it'd be good for science.
It's just a
Something,
stereo camera, yeah.
Stereo.
But I don't want anyone taking up my feeds for next cruise because I want my shadow cam doing
Your
it in
feeds.
real time.
Wow.
I gave
Claiming.
everything for autonomous, so I wasn't in anyone else's way this trip.
Which is great.
But now I'm downloading it all and charging it and feeling the
yeah.
pain.
Well, there's another dark fiber.
Yeah.
There's another empty fiber on the vehicle.
But wouldn't Brennan be having that for his camera system?
Not sure on that. It looked like some kind of a site.
We need to hash that out before I finish my designs.
At that time, they can have another.
Oh, they're getting a new ROV, right?
They said 2028.
2028.
Oh,
I hope that's
jelly out
So
there.
we have to get the
public
specs.
knowledge, you guys.
Oh yeah.
I don't know what you're talking about.
The specs have got
what
to
was
fit our
that?
payload, right?
Arctibedema?
Oh, what was that?
That was Arctibedema. Something before
Well,
that
it
I
had a
couldn't
red
tell.
stomach though.
No, it didn't. You're making that up.
Didn't it?
Okay,
did.
507, stopping at 500.
a Tuscarora?
Jelly.
Another little jelly.
Maybe.
Yeah, it looked more. It looked pretty good in the DPAV.
Graded, was it?
Okay, 500. We got a bottle to trip.
Oh boy.
Oh wow, we can watch it happen.
Yep.
That's amazing.
Oh, lights.
Next is bottle 2.
That's fine.
So we have one stripped already.
Bottle 2.
Okay, go for number 2 and 3.
Just one bottle.
Just one bottle.
There it goes. Oh, you can actually see that switch.
Already.
Yay.
Okay.
An event.
That's so cool.
Get the camera back in front. And
That's a sample of that?
up we go. It's an triples,
Niskin.
trips
Oh,
and
there's a
Niskin bottle.
sample. There we go.
They're mounted on the top of the vehicle. Kitty
And
Panther
let's
fish,
add an
probably fish.
annotation comment as well.
Sample, is it a sample?
Yeah, it's a
I think
sample.
it's a sample.
A
Yeah.
water sample for
Yeah,
stable isotope.
that's good.
So misses.
Jelly,
It's
probably
going
still
to
misses.
find.
Carry on going up.
Carry on going up. Yeah, with some forward velocity.
So
Yeah,
much
angle up.
How many more water samples?
Okay, we are
are
at
we gonna do?
500 meters with
100
45
meters?
minutes
I
to
think the rest are
go.
at the surface. Oh, what's that?
That was, it
Oh,
can't
that's
stop.
okay. It's a hippopotid. That's all right.
Okay, Hippopodius.
Oh, I just hit
There's
There's a bigger
another.
siphonophore behind it.
That's an apolemia, I think.
Apolemia. Okay, drive through it.
At least see the nectar
Go
side.
around it.
You see
It's already
the
exploding.
That's an apolemia. yeah.
apolemia. Okay. Okay, keep going.
Hippopodidae, not sure what genus it was.
There's a couple of those. I don't know.
Apolemia as well, right?
Hmm?
That is the next.
The next stop is at 50 meters,
15.
which will be during the pull process.
Return captures.
Say again?
Return will capture. I just need to go to the.
Oh, you want me to hit something?
Yeah, if you see something cool, just press return. It'll capture a frame, and then I'll annotate it when I get back.
There's something cool
Those
right
are
there.
probably doliopsids. Only 4.
I'm so helpful. There's another cool thing right there.
That's a bug mania.
That was Bargemania? Okay,
Yep.
keep going.
It's a siphonic.
Yep.
Physonic siphonophore. In Japanese, we call it
There's
the
another cool thing.
snake siphonophore because
There's another
it's
cool thing.
2
Seeing
things,
is
I'm not
so-
sure what.
swimming.
I'll do my best, Dougal. If you need to go eat.
No, we just need to go to the bathroom.
Only 15 minutes until midnight dinner is over.
I'm gonna skip it. There's too much going on.
Got this question about vertical migration. Let me put up our EK80 for you.
Actually, yeah.
Russ is a hard taskmaster going at this speed to get up
A little
shallow.
bit more
There's
forward
something
motion.
there.
So we can see the migration layer.
Is that a
Something
silhouette?
there.
Can't really tell.
Not the most obvious right now in that, but basically we can use these to
Probably
track
with the
the
speed.
migrations up and down in the water. And that's one reason these dives are when they are, is we're trying to catch that migration at its high point, so we don't have to go as far down with our gear.
470 meters.
Repel animated.
Oh, that's awesome. This fun.
We calm down a bit now. We're above the. We're above.
No.
So Mrs. Maybe? Or was it a house? One of the two. Something. Call it a something.
Target is what we call it.
That was a fish.
Am I too fast?
It's a little fast. But I don't know if Russ wants us to go that fast.
I need to be seeing big things. Fish, can't tell. Is that an oik?
That looks like an oik
Can't decide if it was an, yeah, probably another oik over on the other side.
Yeah, yeah.
Little scythe.
Yep, and a
There it's-
coelodendroid as well.
Yep. They seem shallower.
Yeah.
Maybe they migrate.
Who knows? I don't know how they'd migrate. They'd have to do it all by buoyancy
Yeah,
change.
They totally could though.
Go fast.
Okay.
Little jelly
Migrate
or a
by
house?
buoyancy
One of the-
control.
Yeah, the sonar I showed was from the Kongsberg EK80 echo sounder. And yeah, it's fun because we can see the migration layers, but also sometimes when we deploy equipment, we'll see it go up and down. So like last cruise, we didn't have ROV dives, but we were deploying various instruments down into the water, and on the monitors, you could see some of those instruments making their way down.
But if you do need to go to the bathroom, I can hit a button for you.
Oh.
Is that a scythe over there?
Yeah, not sure which one though. Kind of cover it,
House. Another house.
So jelly.
433 meters. I think that was a price area.
Yeah.
Jelly, sun type over there. Keep going. Or a house.
Oh.
Scythe?
Yep.
Or just mucus?
I think that was the scythe.
Not to the right depth for squink yet.
You're gonna have another self-sampler tonight?
Another which sample?
Another self-sampler? We had a squid go.
We might.
Yeah.
Did we get some off the vehicle last night?
We had one squid swim straight up the
Okay.
nozzle.
Ah, cool. Okay. He was probably trying to go after the Tomopterid.
That's right. It knew. It knew its mortal enemy was down in there.
House.
There's something way in the back.
Yeah, let's call it a house, something. At this speed, it's really hard because we don't really have the time to look. We've got too much vertical and not enough horizontal. The horizontal buys you the time to kind of for your brain to see something and piece together what it is. And when we get going too far vertical, as you're doing now, there was a shiny and something else on the other side. Ooh, scythe.
Nice
Yep.
scythe.
It was a ooh, what's that? Big out
Looks
there.
like a Keatinga?
Yeah. We've had them come up on the porch.
There's another oik
We've had
house,
Couldn't get that. Up.
whatever that is.
I think that was another silo den, whatever.
And they were like spraying down the RV or something.
There looks like another oik house.
House.
Alrighty then, mateys.
You ready?
The Southern Ocean. We have so much.
Yep.
Probably house, yeah.
Oh, there was
And a
another
shiny.
one of those Phaedarians. Another
Red shrimp.
suggested.
Hmm? Yeah. $400 meters.
So there's a few things for
Oh, there's a
Oh, that
nice
was a house and
Yeah.
I think it almost looked like a fish.
Where's the scattering layer at,
You're
John?
always out there.
What's that?
Another side.
Siphonophore over there on the right.
Okay.
Hard to tell what it
House
is,
or rosarian, I couldn't tell. Another siphonophore off in the distance.
On the floor, fascinating.
Oh.
Can we go a little bit further forward as we go up, please?
Jelly. Shrimpy.
A little bit more forward velocity if we can.
More forward?
John, I don't know how you're taking
House.
dictation.
It's really fast, but I'm not writing anything. It slows me down too much.
Oik.
383
Say, oh,
meters.
no.
It works, I guess.
But I thought that like someone goes back and checks it later.
I don't know about here.
Oh.
Connie's making some huge system that'll automatically annotate this entire video later, right?
She is.
We've already
Yeah.
pulled out frame grabs from our last 2 days of diving.
So Mrs.
Yeah.
Mickey.
If I have to tie bonds.
Oh, Cereboma maybe?
Yeah.
Fish.
What is
Can we
that?
Stop for that.
See it?
Can we go see the fish?
To the right.
Yeah,
Over there.
He's gone.
Might have taken
Or
off.
straight ahead.
There's a nice
Or is that a
jellyfish
jelly?
just behind it. Or is that a jelly, did you just say?
There's a jelly right
Or
there
is that a
too.
jelly? Jelly.
Yeah,
Fish booked it.
straight up.
Oh, that's a shame. We missed the fish.
There you go. You can see it in the other camera.
Yeah.
Hey, John.
That's it. Oh,
There's a
Oh,
nice jelly.
it's a halotrephes.
that's familiar.
Yes.
It's, I don't remember what.
Is that the Bigelowy?
Halotrephes marsi, the one we
Marsi,
caught yesterday.
okay.
Is it a Trachy Medusa?
Yes.
Yeah.
Good name.
Okay. Up we go. Siphonophore. So much of this is just going too fast, ID. $367 meters.
Improvements for next
That's fine. And then
we've got Tyler Summons versus
Tyler's looking fresh.
You are. You look like you're well rested. Wow. There was a 9 a.m. room check, but we didn't know that. It was really funny though.
Risaria.
The whole thing. I felt so bad.
the one that used to be known as a foreign.
Because we were the first cabin.
Did you scream?
Looks like a scythe coming down the side, or maybe an egg case, or the rides area that looks like an egg case.
Ready to defend yourself?
But regardless, if you're bored,
350 meters.
Mark, do you remember when we had like an extra camera on top of Iris?
Top of Iris.
Like on top of that housing, like to help give some sort of orientation.
Next time you're gonna have a super shadow map.
We have them out.
Yeah.
We just, I think we just
Right here.
We've got to work out what Brennan needs as far as payload connectors and things go.
So big assumptions. That was
Shiny rise area.
Oh.
Sitting here, this seems like it wasn't quite perfect enough to get.
Could we maybe angle the main camera up a little bit more so the particles are coming more from the front? Is that okay?
We're wondering.
Yeah, tip the main
cameras
camera
are kind
up.
of reflective as well.
We have a wider field of view.
In the meantime, sending the biopsy sample.
Yeah.
You have
A bit more, Google.
Hey? That's better, thanks.
That's better. Not sure. That was a house.
What's in there now?
It was probably a shrimp. Could have been in that flood. Shiny.
Another rosaria.
Which we can't
We've zoomed once or twice.
Another shiny right there. I think that's what they are.
You're going to send the biopsy sampler to Japan?
Yeah, do you think that, do you have a,
Do I misplace it? Is that how I get my ports for the Ethernet?
do you have
Jelly
like,
back
Do I have an
there.
ROV you could go on?
yes, that's what I'm trying to ask, yes. I know I'm not gonna sleep, but my brain is stopping.
324 meters.
I'm amazed at how few squid there are. Or even fish, really.
Quite high in there.
Tomopterid? Tomopterids have eaten them all.
You really think so?
That are high enough. I don't know. I'm just looking first. 250. That's good.
Not like
So,
No, it's just more like you need something to see the net really well, or do you have?
yeah, I guess there is one like would have been it.
Some kind of a jelly just went by on the left.
Yeah, that one can hit the D's.
House.
Yeah. So the function, like this one might have been used to just make sure you don't hit the D's while moving.
Shiny.
But we also have really weak moving it at all. We're moving it today. And then that one was used for the times that we bring iris over more centrally. I feel like Drew still have some of that.
That was the siphonophore way out in
And
distance.
then we lost the animal once or twice.
And that was a jelly, I think, way out
Pick a
distance.
spot for iris and keep it there, and then we're able to go quickly from one instrument to the next. Otherwise, we just lose the animal when we're like adjusting where the camera is. But if we
Shiny.
were to fix it in place, having I think a camera that gives a wider perspective over the housing will
300 meters.
orient targets in the camera.
Siphonophore.
really?
Yeah.
Whoa!
Oh. You need to see an end?
That's a crane. It's pretty long, isn't it?
Uh-huh.
Another one from South Africa and onward.
Siphonophore just blown apart over there. Small one.
We found one earlier that we were pretty sure was attached to South Africa.
Based on the spacing, it was probably a Praed.
Yeah. Rosacea, maybe. Fascinate other radius, not radius, or rosaria, sorry.
Yeah, it's in here already as well.
Oh, yeah.
All right, so there's some genome sequencing samples in there, I guess, right?
Yes.
Not sure of that.
You shall not sleep.
I'll follow your routine.
Shiny.
We're not gonna get
Another
sequence
area.
tonight. We're all supposed to, but
We've done as best as we can, and what we're seeing.
I'll get them ready.
Which one do you want to go?
Oh, because you recovered? Okay. Wherever you want to put.
You have to be
I was
outside.
asking him if we could send.
What's that?
Do you want to be outside or do you want to go?
Really kind of a strong preference either way.
House.
For the thing walls.
Not sure.
Maybe we just don't like it.
You have to
Gives
be
a big
outside.
problem.
Have to be outside.
Because
Haven't been outside yet.
Like we would in the past one, two sort of ray traced off.
Flying fast.
Shiny.
Now we have one that's different.
Oh, that was a squid.
Yeah. I'll need the
House.
on-the-job training for the verb scores. Okay.
Not sure.
The dive.
That is something swimming and
That's another
yeah,
Halotrephes.
Halotrephes.
Sounds like he knows. Okay. Shoot information next time.
I mean, there's a number of things that we could have done. But not having it. We have enough.
Shiny.
Another Isarian. Alps,
Yep.
Rosarian,
Shiny.
Rosarian.
Oh my god.
House.
Or was it a squiggly, right?
How did we get past the stop?
Yeah, I think it was a house.
Is it a chain?
No, solitary. Shiny.
Interface game values and the livelihood is worth staying up for tonight.
No.
Put it in. Probably doesn't have to.
No, I don't think. It's perfectly fine. Also,
House.
Frank is watching.
Shiny.
Livestream.
Shiny.
Who's Frank?
Frank, if you're there, we miss you.
Who's Frank?
I'm not sure.
250.
Oh, that guy.
Yeah, that guy.
That guy.
House. Shiny. Some kind of jelly.
It's a trend where you show an idyllic
Scythe.
scene you'd like to chat with a friend in.
Yeah, but I don't know. I guess that elevator is something to do. Dreadful. That's what it is. Dreadful.
So misses.
But remember, our socials are way cooler than we are.
Does he have some
Scythe.
Frank is here.
I saw you run over.
240. Did we cross with scattering layer?
Reach ROV.
Scythe. Can we go over that, please?
Yeah. Oh, was there something? A volunteer, I think. Yeah. Volunteer.
I think it was a cyclonic
When
board.
we get closer to the surface.
No.
Roger.
It's too fast.
Who's Mr. and
What?
Mrs. Jazz?
In the middle?
What?
Who's Vijaz?
Scythe.
Did you see it?
No. There's a few species named Vijaz.
Oh, really?
It's very strange. Oh, Medusa?
House, maybe.
Yeah.
Like
Is it a house?
Forscalia.
Yeah, I think so.
Just Sweden,
No, I don't see anything.
Forskull. Forskull? I feel like I'm coming into the middle of a conversation in your brain that's been going on for a week.
Get out.
I'm
Get
trying
out.
to get oriented
Get
in there.
out.
It's a busy little place.
I like that the emojis seemingly get translated.
I know.
Which language sun
Sorry.
emoji was or star emoji is that?
Never know.
Is the data logger just using emojis?
I think that was a Risaria, the spherical ones.
Oh. No. Oh, another.
Not sure.
And that was.
Might have been.
Might have been another Risaria.
Fish per house.
215 meters.
What else was in my notes?
It's kind of the same with the light buttons for iris. If we put the button in, but I also probably don't want it to stay up. For the set all to this power level.
Oh, we're gonna be
So
swarmed
my
by squids again?
priorities.
I don't know, that's what we're wondering.
What do you think that was? It's too fast.
Gotta get out.
Yeah, we gotta get out. What do you think that is?
Is that something you want to target now, or is it something you want to target?
How are you feeling?
Tired. I have a commitment
Hi.
also in the morning with Stefan to fix the
200 meters.
We need to be directly
help us down on top of where we did 3 dives.
Right.
House.
Do that in the morning.
House and couldn't tell what it was off the distance. Might have been a Solstice.
So actually convince him that he doesn't think we can.
What are you labeling that as, John?
There is a house.
Yep, it was.
House. I don't see it now.
So we're gonna get another here. Model there.
It's much better in that where you don't have to see it. Just mark it.
And ideally, I'd have
Oh,
an ass.
what do you think that is?
I don't know what that was.
Like
Oh,
what,
the
John?
fish?
Yes, that would make our life a lot
Yeah.
easier. So that's how NASA's.
They're shiny and transparent.
Here's my network attached storage, which is attached to one computer. So we also have to start copying the
Google. Yeah, there was a self-like chain pass. I don't understand. When I capture, I'm not seeing the contrast that I see there here. Like I can't find it, but I could see it there.
Yeah, I don't know. Because the monitor
Something
maybe
is
set up with different black balance or
Yeah.
something, and I'm not sure what I'm getting in the HDMI feed here, if it's going through multiple systems.
Oh, so there's a
I
new
see.
It just added.
It does seem very grainy, doesn't it?
Right? Yeah.
What?
What did I look at
But
before?
even when there was, see how the
Part
color
one,
balance
part
is.
Part 1 of 903.
Medusa.
Part 2 of. We've looked at 3 total.
Yes.
By the time I go,
That's
it
the
doesn't show.
first and second half. It's dive. The first dive. First half is the
It's all
second dive.
behind.
This is
I
the
was
second
like, I've been
half,
clicking
second
and clicking and clicking, and nothing is changing.
600 some images.
Ah, sorry.
But the inference is running at about 1x speed. So
Click it.
House.
It's orange. You're live.
It's okay.
Flashing
Fish.
on you.
12 hours.
Yep.
There you go.
Wait, how
Nope.
long is it taking? 1x?
We're in the desert now, aren't we?
About real time.
180.
There we go. Radio check.
They got loud and clear, possibly slower.
See, we're in the desert, and most of the time we're towing at this depth then. Like 200, 300. Either we go deeper. Oh, house? What do you think? Oh, on the left, Colidarian?
What is the bathyc pelagic? This is the pelagic where the bathyc body is.
I don't get the contrast here.
Check.
Oh, something is here.
Naka Medusa?
See, I don't see it here, and yeah, maybe you're right.
Yeah, they don't show up a lot of the time.
Okay, Narca Medusa. Narca?
Okay, so it looks like
N-A-R-C-O.
give us test images until tomorrow.
Medusa.
Well, we probably won't have it until the dive size. It's something we can verify, but isn't really agreed that we should do our own testing. I did what I could with some of the recordings.
Definitely more interesting deeper, isn't it?
Missing a second. Wait, I hope we are.
Sped up and go see what's happening on in the shallows.
Like this to make things
Nothing's happening.
You just can't see it.
Oh,
because it just
They're all
feels
small.
like success
Oh.
rate has been
Colidaria?
Was it? I thought it was
worse than I thought we were. Surely more dialed in.
No, Colidaria, yeah.
Looks too wiggly to
Yeah.
be Parson.
No, it's
That's what I
not a
was gonna say.
parasome.
What
No.
is it?
Has no polyps on the surface.
Come on.
Colo-
I thought it was an ostracon.
No.
Am I being
We
ostracized?
haven't labeled any ostracods so far.
It was a female.
Yeah, it'll be in the middle of the dive.
Do you know what the cod is?
Yes, it's in the room where we do the sorting. Oh, another colored area. You know the room where we do the sorting?
Yeah,
We
where it
must
comes,
be coming
where it
above
comes
the
in.
thermocline now, are
We do the initial.
It's also.
Where like
I
the
think that was a house.
solar truck sorting, there's a sink and then there's
It's one of the reasons you see fuel pressure is funny.
Exactly where you left
I
it.
mean, you're right. Remember
So there you go. I'm invented.
All sorts of stuff.
But
Yes,
it's
they're-
in the room
they are
right here,
always
this sort of fairly brief passes, right? There's rare.
Something.
All right, that's just about us at 150. So we'll be moving into recovery here shortly. I'll be bringing the mics down, but we'll leave the feed up until we're on surface and recovered. So thanks for tuning in for another dive stream, and hopefully we'll see you on the next one. Keep your eyes peeled.